just wanted to quickly go over a bit of a historical background about this session. This session in many ways was incited by the Committee on Credible Practice of Modeling Simulation in Healthcare. And between 2010 and 2012, there was a lot of discussion amongst the consortium here that, how do I make my model believable to the clinician or to the end user? And there was a really hard, it was really hard to kind of translate the models into some useful state. So the idea came up that maybe we need to have some kind of process or guideline for ensuring that there's a constant communication between the end user and the researchers, and also a process that's followed to instill confidence in these models. So in 2013, that's when the Committee on Credible Practice of Modeling Simulation was established. And the charge that we were given was to develop guidelines and procedures for credible practice, to demonstrate the workflows of credible practice, establish consistent terminology, so that between the different disciplines, that when we're actually talking, we understand each other. Because the term validation, for example, that can be interpreted completely different between a biologist and mathematician. So, and also to promote good practice, because you can have guidelines all you want, but if you don't actually actively promote them, who's going to adopt them, right? And so the end product of our efforts is supposed to be basically a guideline for credible practice of modeling simulation in healthcare. And ultimately, we hope to come up with some kind of a proposed model certification process. We don't really know what that looks like at this point, but that's one of our objectives. And lastly, to identify research that can actually help advance these two endeavors, because the reality is that as things evolve, we know that we're going to have to constantly adapt to the new methodologies that are going to be coming around. The way we established the committee was we understand that this is a highly multifaceted or multidisciplinary endeavor. So we have multiple disciplines that are represented, engaging diverse organizations, also including a variety of procedures and guidelines and existing standards, and considering that their application across many areas. And our membership, we have an executive council, and then there's an advisory council, where the executive council does the brunt of the work, and the advisory council advises us on that. And each member that's on there represents many of the different disciplines and areas that are listed on the side over here. And it's working towards the guideline development. We are working towards what we're currently calling the 10 simple rules of credible practice in modeling simulation in healthcare. As you can see here, these are the 10 rules. These are just the abbreviated versions of them. If you want more information, there's actually poster number 1 that gives a lot more detail, and there's a specific example to demonstrate how you can potentially work with these. Now, the objective of this session is to have a discussion amongst the new awardees of what their credibility plan looks like right now, to share each other's idea and also see what the overlaps are, and also to generate feedback from the community, because the reality is that as a community, as individual researchers, we have our ideas of what credibility looks like, but we need to have a dialogue to ensure that we're on the same page. So with that, I will bring up the first speaker, and that's Dr. Basanta from Moffitt Cancer Research.
Is this working? Hello, thank you. So I'd like to start again thanking Bluestein and obviously Manana and Lilem and also Jennifer Coach. Without her help, I don't think Connor and I wouldn't be here presenting this. So we basically, this is the first time we come to this meeting, and we're very impressed. We actually learn a lot. I found at least 1 or 2 working groups that I'm more than happy to contribute to in the future. And on Wednesday, you saw me presenting 5 years of research in about 10 minutes, which was a bit of a challenge. And today I have the same amount of time to present the credibility plan, which is a completely different kind of challenge. And the challenge comes from the fact that when we started thinking about submitting to this mechanism, no one in the department knew what a credibility plan was. so we didn't really have guidelines on what a credibility plan is meant to look like. But we came with something, which is what I'm going to be presenting today. So I'm definitely looking forward to some, and I'll just highlight the word constructive criticism here. So here we go. We work in a cancer center, so basically the stakeholders for our work come in three parts: mathematicians, biologists, and clinicians and patients. So basically we want to make sure that what we do is relevant to all these groups, and that all these groups have an input on how credible and how relevant the model is to them. And the way we have thought about the key elements of this credibility plan involves mechanisms. Our models are mechanistic. They are not statistic. They are not about data processing. They are about trying to scan the literature and work with biologists like Lynch, the co-PI of this grant, in order to make sure that we have a relevant selection of mechanisms that we want to implement in the mathematical models. So they are not hypotheses that come out of nowhere. They actually have been presented in relevant publications, conferences, and have been basically studied before, and now they're integrated in a mathematical multi-scale model that integrates all these levels together. The integration and parameterization we think are key elements, because we basically have all these parameters and all this data that comes from the Lynch lab. And the next element is the validation. When we produce a hypothesis, when we want to test something, there is a set of in vivo models that we can use and explore the computationally driven hypothesis, so as to ensure that what comes out of them is actually something that is consistent with biological, even if the model is a biological model that can at least give credence to the results. Translatability, so we work in a hospital, and we are actually talking and collaborating with oncologists, surgeons, physicians of all kinds. So translatability is one of the key things that we think is going to lend credibility to our plans. And open source, in the sense that we obviously, and I think that this is a great community in which to share these things. You guys obviously have bought into this even before we started thinking about ourselves. But making sure that the tools we use and the code we produce and the data we get are released and spread as wide as far as possible. So year one progress is a much shorter list than the one you saw in the previous slide. I think a lot of you would not completely understand why is that. We just started. We have recruited not long ago the postdocs and grad students that have been working on the project. So we have been preparing previous code in order to make sure that it's not just released, but also released in a way that will be useful to other people. We have produced this multi-scale model of bone that I think could be a potential useful tool for the people who want to understand all kinds of things about bone, be it cancer, metastases from other organs, or just injuries in the bone. We are trying to understand seam decay and create a page in which your project will be hosted. And at the moment, we have been very successful and make sure that the model that we have just started developing now is consistent with the data that comes from the Lynch lab. So basically, the model guides experiments, and experiments have basically shaped the model. So we started very, very simple, and we just keep adding things to the model, following what we promised in the grant, in order to make sure that every step of the way there is data and there is biology to support what we're doing. And in terms of challenges and opportunities, I normally just focus on the challenge, but it's true, there's an opportunity here to improve. A lot of the people we work with in Moffitt, especially the modelists, tend to like tools like MATLAB that are not particularly open source or open source friendly. So we're trying to move slowly from tools like MATLAB to the tools that will allow us to share the code with people who don't necessarily have the license to these expensive tools. We really want to make sure that we have an impact in the clinic, and that is a big challenge which still we haven't completely figured out, which is the clinical data can really be easily shared, so we need to be able to compartmentalize, so everything that can be shared, but that will have to be independent from the data that comes from the clinic, which obviously is not meant to be shared. And also, as part of this community, I've seen that there's a number of tools that other mechanisms not usually offer. The wiki is just one of them, but it's just one example. We've been working in understanding the power of these tools and how better to integrate with the MSM community with them. But there is a steep learning curve which is trying to sort of slowly get over. And in terms of the uniqueness, or what we brought to the table of the MSM table in terms of uniqueness, I think this is one of the main things to me, which is that by working in a cancer hospital, in a very integrated department, we actually bring a lot of people with a very diverse variety of backgrounds. And I think that in terms of making sure that this is credible, it's not just a question of finding one technique or tool that would allow you to assess the credibility. It's just having people who actually come from these different backgrounds, all of them have a stake in terms of the usability of the model, in terms of the biology you can discover, the mechanisms in terms of the biology, and the applicability to new treatments or new ways to apply existing treatments that I think lends a substantial amount of trust and credibility to the work we have produced in the past and which we intend to produce now for the MSM. So in terms of third-party evaluation, this is one of those things I learned after the fact. I decided to be honest about what I put here. I know that other people have actually created Consortium, which is external to the organization they belong to, in order to come once a year and evaluate the work they've done. What we wrote on the credibility plan 2 years ago was somewhat different, which is we are in this department, which is it's got 7 faculty, lots of postdocs. So we have people like Sandy Anderson, which is a mathematical modeler, some of you know, Bob Gillis, which works in the imaging department and is well known for trying to understand evolutionary dynamics in cancer, who are have volunteered in order to make sure that both the modeling part and the biological parts from our model actually are consistent and credible and ready to be shared. And also, we have a series of seminars where we present and we get all kinds of criticism. So basically, we believe that if not necessarily sufficient, at least adds to the ability of bodies that are not necessarily part of the grant to evaluate the quality of their work. And I finished well ahead of time, so if anyone has questions, that's fine. And if not, I'm sure that the panel will be an ideal platform for that. Thank you.
Yes, we'll hold questions until the discussion panel. And if you can actually put your questions on the wiki, that's the most efficient way to actually handle them. So our next speaker will be Dr. William Cannon.
Okay, so thanks. I'm Bill Cannon from Pacific Northwest National Lab. On this project is Jeremy Zucker. He's here in the laboratory in the audience too, and Scott Baker, and we're with Jay Dunlap at Dartmouth and Jennifer Hurley at RPI. Our project is about multi-scale modeling of circadian rhythms in Neurospora spora, which is a small filamentous fungi. And so before I get into the credibility plan, let me give you a little in a nutshell about what the project is about so that you can help us guide us through model credibility issues. We're really trying to go from enzymatic reactions. Let me get the pointer over here. Okay, so enzymatic reactions on the millisecond time scale all the way up to the time scale of circadian rhythms on 24 hours. So this is quite a large set of time scales, and we're developing new methods to be able to do that, and the model credibility plan relates to those new methods. And you'll see why when we get down into the presentation, why Neurospora was chosen for this. So why develop new methods for modeling metabolism? Basically, right now, it's very, very difficult to model metabolism using the law of mass action because it's really hard to get the rate parameters. And so what we've done is we've taken a look at the ODEs that are used for modeling, that used to represent the law of mass action, reformulated that from the perspective of statistical thermodynamics. Basically what that allows us to do is substitute chemical potentials in place of rate constants in favorable conditions. And those chemical potentials, because they're measured at steady state, have dynamical information about the cell. So we basically end up changing the reference state from equilibrium to a non-equilibrium steady state when we do that, and that allows us to model a transient non-equilibrium dynamic quite accurately. And it also allows us to use state functions to go between different steady states in the model, the non-equilibrium transients too. And this, I mean, if you're familiar with statistical thermodynamics, this has to do with Jasinski's equality and generalized detailed balance. So the intended use of these is for, right now at least, for modeling things within the cell, specifically metabolism and modeling regulation and protein expression. And so we've basically got two use case scenarios. One where you've got all the experimental measurements that you want, and that would be metabolite concentrations inside the cell. And that's still a pretty tricky thing to get, but a few groups have been able to get some pretty good estimates of those. And also the other type of data that we'd like to have is metabolic flux data. That's also very valuable data. But for most biological systems, you won't even have that. And so we use, in those cases, we use maximum entropy production rate assumption. And basically what that comes down to is using standard chemical potentials instead of steady-state chemical potentials. And that's a maximum entropy production, which you can think of as the least biased assumption that you can make for a biological system. So in principle, the first use case can be used for abiotic systems as well, but not the second, because the concept is based on aspects of natural selection. So with regard to the model credibility plan, we've got two issues here. We've got the mathematical model, which comes down to the parameters that are being used. So how do we justify those parameters? How do we evaluate the uncertainty of those? And then there's the biological model, which mostly has to do with, is the biological model complete? Have we accounted for all the metabolic reactions and all the regulation that would be important? So the uncertainties in the parameters in these chemical potentials will show up in the dynamics, and of course the dynamics will then show up in the wrong timing of the events in the biological model, and especially with the circadian clock proteins and metabolism firing at the wrong time. So we know the link between the two. And one of the unique aspects of this is this maximum entropy production assumption relies on those energetics, which relies on the chemical potentials that we're looking at too. So again, we're in our first year. In the final year, we plan on providing at least two evaluations. One is on the mathematical model itself, just the use of these ODEs for modeling chemical rate equations based on the typical rate parameter-based ODEs. And the second one would be evaluating the full biological model and understanding the math as that scales to a much larger set of reactions. And we plan to supply these in usable forms for the user. MATLAB for the simple mathematical model to test, and Python notebooks or possibly C library for the full biological model. If we supply that in a Python notebook, like a Jupyter notebook, we can actually guide the person through that instead of just giving them raw code and explaining how things are to work. And we've allowed for 3 weeks of funding for a third party to test this, but we've also allowed for bartering within the MSM consortium so that you don't have to exchange funding within between institutions, which is always difficult. And I've talked to a number of people here who I think are fully capable evaluating our model. So the first year progress that we've made, this graph over here on the right is kind of a summary of that progress. We've evaluated parameter space for a set of simple coupled reactions. And the red lines are the results of a kinetic model using rate parameters where you solve the ODE. And the blue lines are the statistical thermodynamic formulation of the same ODE. And they've actually been offset in the top two lines up here by a little bit, just so you can see the difference. And they lie right on top of each other. So we've been able to show that for equilibrium situations, for non-equilibrium situations, for steady state, for non-steady state, for transients, we get exactly the same results using the statistical thermodynamic approach. So when we scale this up to test this on a full biological model that might have hundreds to thousands of reactions, we need a model credibility approach and uncertainty quantification approach that scales as well. So we've been talking to the DOE centers. There's actually 4 DOE centers funded on uncertainty quantification, and they've got a lot of nice software packages that are out there that are relatively easy to use. So we've talked to them a little bit about our approach, and one of the early ideas right off the bat that they agree would be a good way to approach it would do some important sampling where you're doing a Monte Carlo approach to look at your parameters and understand how that affects the fluxes in your model. But there are other approaches too. We heard about Bayesian modeling this morning, and that was one that we actually discussed with the DOE centers too, and that is definitely an approach we're interested in. There's an approach that you're going to hear more about from Mike Henson, I believe, on stochastic expansion to look at uncertainty quantification. That's also something that's very important to us. But also there's a new method came out specifically for circadian rhythms that will allow us to compare the experimental data over a 24-hour time period with the predictions too, called circadian response analysis. And it's kind of a metabolic control analysis for circadian organisms. So this is something that sounds pretty promising. So this is why Neurospora is such a great model to test new methods on, because what you see over here on the left is those are race tubes, and the filament, this fungi goes down there, and you can visually see the changes of the phenotype over a 24-hour period. It lays down these mycelia, and then after a while they create these aerial hyphae, and that marks the end of the 24-hour period. But we've also got phenomenal data sets from Jay and Jen on the proteomics and metabolomics and such too. One of the challenges that we'll have is with the C13 metabolic flux analysis. That's actually pretty hard to do in a filamentous fungi because they don't like to grow in chemostats. They like to form flocks and really gum up your chemostats, but we have a couple backup plans for that. I don't know that our model credibility plan is unique. I think a lot of the uncertainty quantification tools that we've talked about are the same ones that we've talked about this morning. But regarding the crossover of credibility of practice, the areas that I see here that are really difficult is, sure, you can talk about the assumptions of your model, but a lot of times the end user implicitly violates those assumptions without knowing. And you can list the scenarios where the model should not be used, but you can't list all the scenarios. And so a lot of times, end users are pretty creative about coming up with new situations that you never anticipated your model would be used for. So I think, to me, that's where the dangers are. And that's all I had.
Our next speaker will be Dr. Zhou. He will be speaking on the credibility plan for systems modeling for guided bone regeneration.
Good afternoon. As the organizers for giving us this opportunity to talk about our work on the systems modeling guided bone regeneration. Our young, the project is project. So the basic idea was actually just straightforward. Basically, we want from the molecular level using try to find the basic combination of different growth factors to stimulate the stem cell, and then cultivate the scaffold, and then implant it to the broken area. So the final goal is to optimize the parameters and the biomechanical property of the scaffold. So given that goal, we propose this solution in this study. So the first one basically is try to find the basic combination using some in vitro experiment model of the bone growth in 3 space based on the for the scarf loaded with the program the growth pattern release system. So the third one is basically to optimize the parameters of the scaffold and the no variety in the in vivo system. This is the idea. So we propose this kind of system with 4 components. The first component is the simulation of the growth pattern in the scaffold. The second one on the left is this angiogenesis component. The third one is that stem cell lineage component, and the last one is the bone forming component. So based on that system, we propose the different kind of modeling, such as the pre-vascularization, cytoplasmism, mechanical stress, 3D modeling, and optimize the geometry and the biomechanical property of the surface. So this type of model can be used in a different kind of studies. In terms of the key components of the model credibility plan, so the first one is about the variety system in vivo. That's very challenging. We will talk a little bit later. And tested by the third part, because I came from the Wake Forest, so we have one team at the Wake Forest Regenerative Medicine Institute. So they are going to independently validate our model, and also one team at UT Hills, they are also going to validate this model. So in terms of the accessible of this process, we just checked with our program directors, and we hope to have some spotlight and also cover some workshop in the orthopedic conferences and also other conferences. So in terms of the timeline, so we plan to because the temporal large-scale modeling done within the first couple of years, and then the 3D model in the second couple of years, and also validate the system in the last couple of years. So for the partner study of this project, so you can see from this starting from the stem cells limited by different growth factors, we collected the data at the genomic level, protein level, and also the cellular level. So we integrate this data together to understand the downstream mechanism of the different growth factors and try to find the correspondence between these growth factors. In that way, we can use a model to model this whole procedure and eventually predict the optimal combinations. So that's we got the prototype of this model and also validated by this model. Some results I have given. And also meanwhile, we did a lot of Western PCR and logout experiments to verify the discovery in this study. So here is the very simple discovery from this study. In this case, we found the two most important passes, and one is under the beam 2, and another one is also under the IG1 to stimulate the differentiation of the at the different stage of the stem cells. So here is many in vivo experiments done in Dr. Young's library. So basically, already showed this kind of a progress of the bone growth. Now the issue is how to model this kind of a bone growth with some pre-vascularized scaffold. So yeah, here is one result simulated from our model, which is basically use our model to predict the release of the different growth factors under different pore size of scaffold, and you can see validated by the experiment results with the V2 study. So in this model, we can also predict the effect of pore size and the polar site based on angiogenesis and the bone formation, as well as the combination of different growth factors, as well as by considering the neurovascularization with or without mechanical stress consideration with the scaffold. But this needs further validation, especially in vivo situation. So in terms of the challenges and opportunities with this model, so as you already see, a lot of our study were finished based on the in vitro experiment. If we move to the in vivo experiment, number of parameters experiment need to be changed, and also in the in vivo environment, we have to consider the interaction between the implanted scaffold with the normal tissue, and meanwhile, the immune response is another challenge issue we have to consider in the future. Another big issue is the translation of this type of model to clinical. In the past couple of days, everybody talked about this issue, so we face some challenges. Hope we can find some new strategies in the next few years. So we do have needed to optimize several factors simultaneously in this model. As you already see, we have a full component and we have a number of parameters in those models. So we hope that we can first optimize each component and then get some initial parameters, and finally we put this component together and then we conceal this component and finally to get an optimal parameter estimation. And meanwhile, our Yannis library is developing some very new Scarfold and materials. So basically, it's called a soft hydrogen and rich Scarfold. So given this kind of new materials, so we are also going to modify our model to basically reflect this kind of new design. So finally, I would like to thank my collaborators, Dr. Tan, Dr. G, and other doctors. Of course, I also my co-PI, Dr. Yang, and his group, and the major funding resource from MSM. With that, I would like to thank you for your attention.
Our next speaker will be Dr. Terry Sanger, and his presentation will be on stochastic dynamic operators and the credible plan for that.
Thanks very much. I just noted to some people before that in an environment that awards funding to transformative research, it may be more important to have an incredible model than a credible model. Nevertheless, I will talk about our credible model. In fact, I am going to probably talk more about validity than credibility, pointing out that credibility is more in the eye of the beholder. So the system that we're looking at is based on a mathematical formulation that I developed. I'm working with Simon Giszter, who's doing the experimental part of this. This is the mathematical part, which is based on the stochastic dynamic operators. And these were developed as a way of describing the behavior of neurons deep within the motor system. And so the question that comes up is if you have a single unit microelectrode recording from a neuron somewhere deep in your brain and you think that has something to do with movement, how do you describe what that does? And the kinds of reasons that this is a problem is because that neuron might fire and lead to a change in the length-dependent force of a muscle, right? So it could lead to a change in a stretch reflex. It could modulate things below it. Or it might fire and change the overall dynamics of behavior in some even more complex way. So the recognition that the firing of motor cells doesn't just cause a force in the outside world, it causes a change in the dynamics. And what I mean by dynamics is the relationship between sensory input or sort of state of the environment and the change in that state that occurs following the neuron's firing. So the stochastic dynamic operator sort of generalizes the concept of either a spike-triggered average or receptive field into a dynamics framework. And essentially what it does is you can think of it as a state-dependent spike-triggered average, right? What would happen to the force generated by a muscle depending on different lengths of different rest length of the muscle or what was happening at the time and other variables. And you can use this also, the same formulation, to look at the linkage between different neurons and different populations of neurons. Roughly, we describe these things. This work. Oh well. This might work. You see that? These things can be described as matrices, and they sort of have, the input is along the bottom of the matrix, and the output is along the vertical component of the matrix. The density of the matrix just sort of says if I start at, so if my input value is sort of 40, if that's sort of I'm at 40 millivolts or something when I of maybe EMG or something when I start. And then after this neuron fires, the hot spot here, the red zone, says that after after that thing fires, maybe it'll be 60 millivolts after that thing fired. But if it didn't fire, maybe it would have stayed at 40. So it basically gives a stochastic description, a probabilistic description of what happens in all states and what the likely states are at the output of these things. But the nice thing is if you formulate it, you have some nice properties. You can combine them linearly, so you can ask questions, for instance, if I had a large population of these. So if I characterize one neuron and I say what dynamics does it cause, I could say, well, if I had 1,000 copies of that neuron, what would that do? If I had a neuron of a different type and I had 1,000 copies of that and I combined these, what would they do? If I deleted every other neuron through injury, what kind of effect would that be expected to have on the dynamics? So you can make predictions from internal measurements without really having to know very much about how that neuron created its dynamics, because again, we may be looking at things that are deep within the system and we don't know every synapse between that and the periphery. So it gives you general description dynamics and allows you to look at population effects. So I'm going to talk in a second about the validation, but what we have done so far is really one of the first steps of the validation. This is a new mathematical framework for describing the behavior of neurons and motor systems. And so just the fact that I can get a mathematical result, which, you know, take data and apply math to it, and I'm going to get something, right? It doesn't mean it's valid. So we took a model system developed by Ilya Rybak's group of frog spinal cord. And this is a known model system. So in other words, there's a software. It can simulate the firing of groups of neurons, within the spinal cord. And we can calculate essentially these stochastic dynamic operators for each of those neurons. And we know the ground truth. We know the connectivity in this network because it's just a MATLAB network, right? And then you can see from those, can you reconstruct the expected connectivity within this network? And the answer is yes, you can to some extent. Again, it's a probabilistic description, so it's going to give you probabilities. But it'll give you some idea of what the links are between these things and what are the functions of the different neurons. And as you perceive, the bottom of this thing this is sort of the primary motor neuron level, and we're basically trying to predict EMGs off of this, and we can do a pretty good job of that. And you can predict what happens to EMGs if you delete certain, or you delete or kill or stimulate certain elements within this network as well. So you can look at the inter-network connectivity, you can look at the effect of different neurons on the periphery, and again, we can do all of this in the context of things where there's sensory stimuli being applied. Now, of course, what we really want to do, this is just the first step, but we want to get to the point where we can do this in a biological system. The biological system we're using is the frog spinal cord, and that's Simon's research. And as we get data from the frog spinal cord, which is of course a much more complicated system, this has a total of 8 neurons. Well, depending on how you count it, somewhere between 8 and 30 neurons in it. But of course, the frog spinal cord is much more complicated, and we don't know all the circuitry of that, although this probably describes the major features of it. So we now want to test this in a real system, right? And we'll do exactly the same experiments in the real system. So the proposal for validation involves essentially validating against a system where we know ground truth. And so that's to ask the question, can this actually, does the technique actually correctly identify a model of the type we're looking for, right? And then the question is, the technique applied to a real system, does it give us meaningful results? That's another. So is this something useful? Does it make predictions? And again, the output here is, can we predict the EMG of the frog during certain behaviors in response to certain types of perturbations? And the reason this is multi-scale, of course, is because we can look across the scales of the overall behavior, say a wiping reflex of the frog or a withdrawal reflex. We can look at the population statistics, so we can look at what would happen over large groups of neurons doing this, and we can't record from large populations, but we can predict what the effects would be. We can look at the circuit connectivity, as I just showed you. There's some ability to decipher the circuit connectivity. We can look at the individual neuron firing, did the neuron fire or not, and then we can look at the description of that neuron in terms of its statistical description as a stochastic dynamic operator. So these are the levels of a multi-scale model, and every one of these levels needs to be validated. And the advantage and so now validation of the multi-scale model involves validating the scales against each other, which means that you need to be able to predict from one scale to the other, and to the extent that you have measurements at these various scales, right? So we can validate the stochastic dynamic operators against the known connectivity, right? And that's the step we're already doing. We can try to predict the neural firing from the behavior. So you can look at the behavior of the frog. Did it move? What are its actual dynamics? We can predict which neurons would have fired under those circumstances, and then we can ask, did they? Right? We can try to predict the behavior from the neural firing. Go the other way. Let's measure the neural firing, or try to stimulate the neurons, and see what happens to the dynamics of the animal. Right? We can predict the individual neural firing from the population firing. That's harder because we can't record the whole population, but you can record other cells. So if you're recording 4 or 5 cells and you say, okay, can we predict one from the behavior of the others? Because just as I can use the SDOs to link from single neuron spike behavior to EMG, I can also use it to link from single neuron spike behavior to the spiking behavior of other neurons, right? It's just a statistical description of input-output relations throughout the system. And then you can do the predicted population firing from the known connectivity of the population. So again, the credibility or the validation of this is about pulling together. You have to get the multi-scale levels to work. You have to predict from one level to another. You have to validate this against the data that you can measure. And then it has to be meaningful in the sense that we have to believe that we can extract from this a model, from all the statistical descriptions, that we can extract a model that actually tells us something about how the connectivity, how the computations are actually done. All these predictions, of course, these are all statistical analyses. These are being done in real frogs as well as in the simulation. It's all done with test set validation, of course. We can also do subset bootstrap validation, all the usual statistical techniques. And we can do sensitivity analysis in the sense that you get the same SDO if you shuffle these things in a variety of different ways. How stable are these things over time, and aspects like that, which are important components of validity. And then we'll test on multiple data sets, again, to say, does this apply to different situations? So there's the primary data to be captured, which is the frog spinal cord data, and that's part of this project. There are published multi-neuron data sets, which we may be able to work on as well, assuming that they have the adequate data, and some of them seem to. And then I'm expanding to, because I run a deep brain stimulation program, I have access to multi-channel single-unit recording in awake-behaving children, and so we can do the same thing with them. And so if we get similar kinds of results and similar kinds of interpretability across multiple different experimental systems, including artificial systems where we know ground truth, then that becomes our definition of, or our approach to establishing credibility and validity for the model. So thank you very much.
Thank you very much. Our next speaker is Dr. Michael Henson, and he will be multi-scale modeling of mammalian circadian rhythm.
Okay, thank you very much. So I followed pretty much the format I was given. Before the meeting, we were given a certain number of slides, what to put on each slide, so that's exactly what I did, and hopefully it's on the mark. I feel like all of us may be flying a little bit open loop in terms of, we hadn't seen this before, so this is, I think, a good thing for us to do. And certainly, I think it'll be easier next time because people have a better idea what it is. So this project involves myself. I'm a bioengineer modeler, I would call it. Erik Herzog, who's a circadian biologist, and Yannis, Kevrekidis, who hoped to be here today but couldn't make it, who's basically a computational scientist, I would say. So I talked about the model, so I'm not going to really spend any time talking about exactly what the model is. I'm going to talk about credibility, usage, validity, things like this in the model. So this is what we're hoping the model will be able to predict ultimately. So the properties of the connectivity of the neurons in the circadian clock, the synchronization behavior, in other words, how these clocks coordinate their rhythm, and also how this gets entrained to the environment. So it's not similar to what Phil is doing, a much different system, and I think also different approaches as well. So certainly multi-scale, because the model connects the genes and the ion channels at the individual cell level to the behavior of synchronization and entrainment of the entire tissue in the SCN of the brainstem. So certainly multi-scale. Some dynamics are on the millisecond time scale, some are on the daily time scale, so very, very large time and length scales. And finally, we want to develop some general techniques in terms of simulating these models, doing model reduction, also simulation, not only of heterogeneous sets of neurons, but also very heterogeneous network topology. So these kind of people I thought would be interested in this, circadian biologists, people that are in computational systems biology, people that are interested in complex networks, and also people, generally speaking, interested in advanced computation of the network. We were asked to say, what are the key components of our model? So in this particular framework, we're trying to develop a very modular structure where we have a single cell model, we have a different, let's say, piece of code, if you will, for the coupling of these neurons, which ultimately end up being a multicellular model. and then Kevrekidis' group at Princeton is going to be working on these network reduction simulation tools. So we envision these being different modules where, in principle, you could swap these out if you wanted to. We like to use MATLAB, and we'll ultimately switch this over to SBML. And when we wrote this proposal, there was this discussion of third-party evaluator, and we actually found one ahead of time. And in the budget, there's money for this person to actually do some evaluation work. So this is Stephanie Taylor. She has a lot of experience with circadian models. She did her PhD in this subject. But we're certainly open to other people participating in the evaluation process, too. So this may be viewed as being very good or very bad. I'm not totally sure. These are some of the metrics we thought we could judge the model. And these are going to be judged versus data that Erik Herzog can collect. So he can collect data on individual neurons and the tissue-level neurons as well. So we're interested in looking at things like how fast the neurons synchronize, how we can entrain these to light-dark cycles, and also the effect of different pharmacological agents that can affect the clock. In terms of the simulation tool, really there we're interested in basically speeding up the simulations because we'd like to do lots of simulations of different network topologies, their flexibility, and also integration with other code. This is really onerous. I looked at it a few minutes ago. I'm like, okay, they're not going to hear all the details of this. But it suffices to say we have a detailed plan for each year about which module we're going to develop. Initial development will typically be in MATLAB, then we'll transport it over to SBML. Each year in the summer involves an evaluation period where the third-party evaluator will take particular pieces of code and run through some paces. Yeah, so if you're interested in this, we can certainly chat about it. This is in red because Yannis was supposed to edit this, but he didn't, and I didn't modify it. So it doesn't mean it's something super important. It just means I forgot to change it back to black. This project is like several of the projects I think here. It just got started, so we don't have a massive amount of actual results to present. We're working right now on developing and improve. We already have done this type of work before, so we're not starting from scratch. So we already have a single-cell model we're working with. We're looking at improving it in terms of improving the signaling, the gene regulation, as well as the electrophysiology. We are in contact with Dr. Stephanie Taylor at Colby, and she's going to be starting to do some evaluation this summer. And again, particularly for the people in the computational neuroscience working group, if there's some people interested in potentially looking at this, we'd be happy as well to work with them. In terms of network simulation tools, Yannis Kevrekidis already has tools developed, of which Tom is in the audience somewhere. I see him now. Tom Bertland is here from Yannis's group, and he could give you some details on these. And Stephanie will also start to look at those this summer, and again, very open to other evaluators. So we were asked to ask, what are some of the challenges that are maybe specific to our particular project? Well, one of them is that we like to do things in MATLAB, and Yannis's group likes to do everything in Fortran, so we're going to have these kind of maybe mismatched codes in terms of languages we work on. This is just more of an excuse than anything as to why it's going to take us a little while to get started. I just have a first-year student who is just getting started on this. And certainly, again, other evaluators would be nice. We see a lot of opportunities for collaborating with other particular projects. So, for example, Bill and I happen to know each other. So they're working on different system with different tools, but I still think we're both working on circadian rhythms, so there should be lots of opportunities there for collaboration. Also, with people that are in the working group on computational neuroscience, there should be a lot of opportunity there for collaboration as well. And I think ultimately there would be an ability or possibility of establishing some guidelines for how we do these type of neural computation. That would be something potentially interesting to talk about. We're asked what's unique about what we're doing. Well, I think somewhat unique about ours, I mean, not totally unique, is we're going to try to pursue this modular structure. So in principle, if it's standardized, people can swap out different pieces of this to look at different coupling strategies or different network models or neuron models. We're going to try to use what I consider standard tools, at least in my world, which would be MATLAB and SBML. We do, I think, again, have a chance to work with other groups in this particular working group. And I think as far as I know, we're the only group that already has a third-party evaluator that actually is budgeted for. So that's probably unique, although I'm not sure it's good, but it's certainly unique. So we're asked to also talk about how what we're doing might relate to what you're a committee, not a working group, right, on credible practices is. And so what I listed here is kind of, if you notice, unlike some of the projects, you don't see like a direct clinical application of the work, and that's because I don't think in the real short term it'll have direct clinical application, but I think in the long term it will. So what we're trying to do in the short term, which is the end of this project, is to evaluate techniques that we can apply benzodiazepines to basically modify people's sleep schedule according to when they have shift work. And this is going to be done in mice, right? So this is hopefully a proof of concept that we can actually use a model to kind of optimize when these drugs are applied to optimally shift the clock in mice. And then ultimately we'd like to apply some of these techniques in the clinic, which would be, for application of things that have to do with disruption of circadian rhythms like mood and sleep disorders and metabolic diseases, and there's actually lots of them. But sleep disorders, then it would probably be the first one. But, I see this first goal as a 4-year goal, and the next one would be like our next proposal. That's how we think, right? Okay. So if someone's interested in doing this, these are the kind of capabilities, or at least interest, would be nice. It'd be nice if someone had some experience with circadian systems and neural computation. Particular interest in coupled oscillators and neural networks would be great. Some experience with the, model reduction techniques that I talked about on Monday that's going to play a big core in terms of developing simulation codes. Experience with different platforms we're going to develop in the code. And then, typically, because of when we're funded, we imagine evaluation being done at the end of, or during the summer of each year. So people that are, everyone's free in the summer, right? So that shouldn't be a problem. And then we're also asked to ask some questions. Maybe this will speed some discussion in the panel, I don't know. But this is some of the kind of things I thought about that were questions I had about this. First one was kind of obvious one, if there's some kind of collective experience or individual experience of these kind of model credibility plans. Because, some people think model credibility is credibility or usefulness or validity, and it's a little different for everyone, I think. What's the best ways to establish metrics for the credibility of the model? Experience in integrating different software tools or components of different software tools. Best ways to work with the third-party evaluators, and, because we don't really have any experience actually doing that up until now. Maybe there's existing efforts that people are aware of within the consortium that we could leverage. I'm not aware of any, but maybe there are. And then also experience with model dissemination, because I think that's always a challenge we face, is if we develop these great things, how do we get people to use them? I think that's it. Thank you.
Speaker would be Dr. Jennifer Linderman on multi-scale systems pharmacology approach to TB treatment and credibility plan that's associated with that study.
Great. Hello. Our group was funded just starting in September. We didn't get a chance to talk on Wednesday because I wasn't able to arrive till Thursday, so I'll give you a quick recap of what the project's about as well. There's 4 PIs on this grant, so I think it's worth noting that 2 are experimentalists and 2 are modelers. So Vera Dartois is at Rutgers, and she works especially with rabbits, and Joanne Flynn is at Pittsburgh, and she works especially with monkeys. And Denise Kirschner is a mathematician. She's at the University of Michigan. I'm an engineer at the University of Michigan. All right, so our project is looking at tuberculosis, which is a pulmonary disease resulting from infection with Mycobacterium tuberculosis. And the pathological I'm not going to try to point and do this mouth, okay. The pathological hallmark of TB is granulomas, which form in your lung upon infection. That's where the battle is going on between the immune system and the bacteria. And so the immune system is playing out there, but it's over multiple length and time scales as well as multiple compartments. And so the diagram on the right shows in the lung, there's molecular, cellular, and tissue level things going on. The granulomas are these dense collections of cells that form in the lung tissue. But then also there's involvement of the lymph node where the immune cells are generated, as well as the blood where cells are trafficked back and forth. So it's multi-compartment, multi-scale. The things happen at the second to minute, but also over lifetime of the host, which is the infected person. And so this grant is about improving antibiotic therapy for TB. So the granulomas, where the immune cells are clustered together with the bacteria, actually form a barrier for antibiotic penetration, so that's a problem. There's also a lot of new antibiotics coming down the line and repurposed antibiotics coming down the line, and we don't have any sense of which ones are going to be good for TB. And so improving antibiotic treatment for TB, which right now lasts 6 to 9 months as long as resistance doesn't develop, and is fraught with some difficulties in compliance and doesn't always work. Often does, but doesn't always work. We have to remember a third of the world is infected with the bacterium that causes TB. So improving it is going to require consideration of these existing new and repurposed antibiotics. It's a huge design space. They can't possibly test all the different combinations of drugs in clinical trials or even in an animal model. It's going to require consideration of antibiotic pharmacokinetics and pharmacodynamics, PKPD, drug distributions in granulomas, development of resistance, and what would be some of the readouts at the host scale. So in our proposal, we described improving TB treatment by pairing computational modeling of their action of antibiotics together with experimental methods that image the drug distribution within granulomas and also the drug in the blood for PKPD kinds of studies. So the specific aims are to determine spatial and temporal distributions of TB antibiotics in the granulomas and predict the development of resistance. Second, to identify optimal antibiotic treatment regimens for TB. I told you it was too difficult to to examine all of design space in the animal models. The computational models are also complex enough that we can't even examine the whole design space in the computational model. And so what we need is some optimization techniques, ways to search. And so we're working on that. And then also to perform virtual clinical trials to test the treatment regimens that we identify as optimal or better, and to test the optimum one in the NHP, that's the monkey system, against a standard regimen. So understanding a little bit about what is in the model, the granuloma model, which is shown at the right, has partial differential equations, which describe diffusion of cytokines and antibiotic, ordinary differential equations, which describe receptor-ligand interactions and any signaling pathways that we're modeling at the time, and then the agent-based model, which describes the cells moving and interacting within the lung and forming the granulomas. And the bacteria are also agents, so each cell and each immune cell as well as each bacteria is tracked separately. This is paired with antibiotic pharmacokinetics and pharmacodynamics, and the immune cell dynamics in lymph nodes and blood. And also with uncertainty and sensitivity analyses that we've developed over a long period of time to work with these hybrid models that have both deterministic and stochastic components, as well as optimization algorithms, those we're getting at the moment from MATLAB, and the virtual clinical trials. So right now, that means it's about 30,000 lines of code. That's with all options on. I think that sounds a little scary, but you have to understand that, sometimes we've done a detailed investigation into a certain signaling pathway, and so that's there, but we can turn that option off, for example. Several thousand lines of Bash, Perl, and Python script code, 3 open source libraries, and then some of these high-performance computing resources that we have access to, both Exceed and NERSC, as well as some University of Michigan resources. So we were asked to take a look at the committee's recommendations in terms of credible practices of modeling, and so we saw those earlier, so I'm going to refer to these in the next slides, just as rules 1 through 10, and try to link what I'm saying to those. So the context of use, what we want to do is identify immune and bacterial factors that influence the course of infection, and we want to understand how the antibiotic PK/PD properties, host variation, dosing schedules, and things like that affect the efficacy of the drug treatment. And we want to be able to interpret the animal data, which generated in this grant will be rabbit and monkey data. The audience is TB researchers in various settings, and the modeling community, the users, are our own graduate students and postdocs, and via collaboration, experimentalists and other modelers. And the dissemination at this point is primarily through publications and presentations. And I think if we can get this to work, if we can show that we can find better antibiotic regimens for patients, then I think it could transition to being used by clinicians or people designing clinical trials and things like that. But that's not the focus of the years of this grant. Components of the model credibility plan, there's 3 components. The first one has to do with model validation and calibration. So remember that 2 of the PIs on the grant are generating data for the work we're doing. And so we have data on things like granuloma composition size, CFU, which is bacteria, structure, knockout and depletion phenotypes, antibiotic PK/PD, penetration into the granulomas, immune cells in the blood, and so on. So we've got lots of data. Some of it is spatially rich and temporally poor. Some is temporally rich and spatially poor. And we also have new and existing data. So some is being generated now, but there's also some from these labs that's already been published or that they have but not published, as well as a little bit in the literature. So we've got actually quite a bit of data. It's very disparate types of data, and that's one of the advantages of the model, to try to use these different kinds of data simultaneously. The modeling students work directly with the experimental students by email, Skype, and in-person meetings. So we meet together once or twice a year, and the modeling student sits right next to the experimental student and can say, where did that data come from? How did you actually do that experiment? And the experimental student can say, what is that model predicting again? That doesn't make any sense, right? So I think that next to each other discussion is really important for the model validation and calibration. We do a lot of uncertainty and sensitivity analyses with some of the techniques that we've developed in our lab, and we always talk about the model limitations as a frequent comment in the lab group meetings, so we're pretty upfront about that. So we have lots of data to compare to. But the other part of the credibility plan is verifying that the computational code is working as we think it does. So we have multiple users testing under a variety of conditions. One of our former students is in the audience here. He now works in Sasha Poppel's group, and he said that it took a long time to get to know the code and really get up to speed on it, because it's a lot of testing that you have to do as a brand new student to make sure that the model is performing as you would like it to. With the ODE models, we compare in-house and MATLAB solutions for various test cases. For the PDE submodels, we compare to COMSOL solutions for test cases. We also have implemented 3 different algorithms within the code to solve PDEs, and so we can compare. We didn't do that because we thought it was fun. We did it because first we did a simple one, and then the next person came along and said, oh, that's probably not as accurate as we like. Let's put another one in. And then the next person came along and said, oh, that's not as fast as I'd like. I'm going to put another one in. But that gives us great capability to compare, make sure that all are giving the same solution. We use techniques we developed for tunable resolution, so we compare the model the behavior under various coarse and fine graining of particular pathways and mechanisms to make sure that everything is making sense. And of course, we use version control and lots of documentation. The third component of our model credibility plan is the least worked out here, and it jives with what other people have been saying here, and that's an independent evaluation. So the kind of capability we would need is for agent-based models, ODE, CDE, C++, or object-oriented programming. So there are some groups here I know that have those kinds of capabilities. I'm thinking of like Shayn, Peirce, and Sasha Poppel and Gary Yon's groups, but I'm sure there's others. So we would love to identify who that community evaluator might be, how we might work with them. Let's see, we're 6 months in, so we've got a lot of the model up and ready because we have been working with it for a long time, a number of years. And so we actually have things that could be looked at by a third-party evaluator already. We've already got 2 antibiotics in there and published, and we've just put 3 more in, and that paper's under review. So there's a lot of things that could be looked at right now. And so I think I see my time is up. So really the last point is the most interesting one. I think the best way to identify a third-party evaluator, is it the best way to go? How do we work with them? Do they come to Michigan? And so maybe that's something that we can talk about in the panel. Thank you.
Yes. So I'd like to invite all the speakers to come up and join the panel. Joy Koo will kick off with some recurring questions that have come up. And anybody else that may have a question from the audience, I would strongly encourage you to make post your question on the wiki, and we'll try to prioritize the wiki questions. Otherwise, just walk up to the mics and we'll take your questions one by one.
First of all, thank you all for being the first cohort to actually share and do these credibility plans. I know several of you mentioned it's been a little freeform, and I think it's actually an advantage to get the various perspectives on what you think credibility is and what you think a credibility plan involves. I think there were several themes that came up, and maybe we'll start start with one of those, and I think several of you mentioned kind of this third-party evaluations, and I think there are several details around how that might work. How would you, what sorts of instructions would you provide to them? What would a third-party evaluator actually need to really evaluate your model? So perhaps if we could have whoever wants to speak up and give some thoughts about how you think that would actually work and what you think would be beneficial for your particular project, we can start there.
I'll go first. So I think it would be really great if somebody came to our lab for a couple of days and was able to sit with the students and our computer programming person and us and really generate a lot of different questions and share ideas that way. I see it almost as a little bit of a collaboration where they might have ideas of how to do things better and we could go back and forth and talk about that. So in my mind that seems easier than handing our very large code to somebody and trying to get it to work on their machines in a short period of time. But that's just our thought.
Yeah, I think for people who wrote the proposal, we know this, but for the other people, you probably don't know this, is that we actually budgeted for this effort. So there are funds to support the third-party evaluation. So it's not like asking you to do work for free kind of thing. And I do think that doing this evaluation through particular working groups would be a good strategy, because supposedly our working groups are organized around the things that we're doing. So in my case, computational neuroscience working group would be great to collaborate with.
I would think that the challenge, again, it depends a little bit on what credibility means. Validation, we know how to do statistically, and you can certainly have people look over how we're doing it if there's any concerns, but most of us are good with that. It seems to me that there's this model utility, which is a component of this. In other words, is it useful for anybody besides ourselves? And that is hard to, in the middle of formulating a model, it's a mess. Well, maybe everybody else's is. My code is a mess, right? And you're trying this thing, you're trying that thing, you have little leftover pieces, and I noticed somebody was programming in Fortran, warms my heart. And I think it's, and to get that packaged in a way where you can give it to somebody else, and because utility is destroyed by making something incredibly difficult to use. So simplification, documentation, packaging, and then essentially seeing if anybody else finds it useful. And to me, that's the most important thing. Do they actually implement it and make it happen for their data sets?
Yeah, I guess I have a similar opinion with you. So the one challenge is really about the validation in vivo. I guess we discuss this kind of problem all the time and see how can we really apply the model to some animal study. So we also just had a chat with our program directors. We basically, if the model is here, how can the clinicians and other investigators, can really make use of this model and make it really useful for them? Because if we just simply package our model, codes, software together and give it to them, they couldn't understand at all. So I guess there is a kind of, huge amount of communication, or through some hands-on workshop, hands-on meeting to explain the model and how to really apply it to some real applications.
Hi everyone. I did not speak, so I'm Peter Young from Stanford University. So myself is bioengineer specialized in biomaterials and medical device for musculoskeletal tissue engineering. So as a materials guy, actually I saw the value of the computational modeling. And so for us, so when we design experiments, so we are struggling. For example, we try to promote bone regeneration or muscle regeneration or tendon regeneration. So you have so many combination of the signals you can put there. So what exactly put there? So what dosage, what kind, what timing? I think that's where for us it's a forbidden task. But the collaboration with computational modeling guy will be extremely valuable. However, for the third-party validation, so myself as an experimentalist, so I would like to say every model is built on the assumption of some simplicity. So in order to promote, advocate computation, modeling, utility. So I think the best way we put out there. So when the PIs or the co-PIs think this is mature enough, just put website, let the users free download, and also put a very clear sign. This built on the assumption what, or for example, if I provide the data for the bone regeneration, so I have to very clearly say the data I generated in vitro and in vivo based on so what material I use, what property I use. So if you beyond that, definitely will be good if you because the value of the modeling, because we try to predict, try to save the money, save like all kinds of conditions, right? So, but we have to be very clear the limitation we have. So I think for third party validation, just put out free, put on website, allow the people to free download, just test themselves and give you feedback. So I think that's very good for the renewal when they come to grant. Thank you.
Joey, can I interject in a follow-up to that last point? It just occurred to me, though, one of the requirements of this award is also a data management plan. And we never really connected the model credibility plan with the data management plan, but what you just said is about how you organize your data, your metadata, even your, how you organize the model, if whether or not that could be something that could be shared with a third party as well to show how clearly you've, developed your model. I think somebody mentioned something like a Jupyter platform in terms of the model development process, and so I wonder if that would be something useful.
Grace, I think you hit on a point that occurred to me during these talks too, is we have a model credibility plan, but I didn't think to put in a data credibility plan, and that can be limiting the validation of the model. Because I know working with a lot of data over the years, especially omics, a lot of times it's not what it claims to be. And so I do worry about comparing models to data. You may be making a correct prediction. This actually happened in the molecular dynamics field, making a correct prediction, but the measurements weren't really what they thought they were. And so there was a disconnect. And, of course, initially it was blamed on the model, but it turned out to actually be the data. And I know with some of the omics, there's definitely issues like that.
And I would just comment, building on that, is the idea of you have a model and you're validating it against one data set. If you're able to validate against other data sets, I think that leads to the increase of the credibility of your model. And so kind of building on that data management idea is if you are willing to share your benchmark, your data set, that could potentially become a benchmark for other similar types of models.
To add one point on that, so as an investigator myself, I did not suggest add more layer of work on us. So basically, I say this is an ongoing project, right? So we cannot expect just 5 years we solve this like a modeling for the growth factor delivery window. That's a holy grail. Maybe end of my career life, I could not achieve that, but towards the right direction. So that's what I mean. I did not mean I don't not layer more paperwork on us. Thank you.
Yeah, one of the things that I've tried to do, and I do generally encourage my students to do, is when you publish papers, you can publish the data and the code snippets that produce each figure in the paper. And that doesn't necessarily mean putting all the data together, but at least allows people to replicate that and to know, more importantly, to drill down into exactly how you generated it. That's not always possible if the code happened to be, I don't know, TRS-80 assembly code or something. But you may, in a lot of cases, it will be possible to make an extracted code segment, an extracted data segment that can easily be uploaded, often as addendum material to journals.
Gary?
So I have to admit that I was really an absentee member of this committee for the last year, so I'm actually not exactly sure where the whole credibility definition has evolved to. But I think there was at least some attempt to separate the credibility of the process by which you develop and use the models from determination of validity, which is more of a scientific question. And I think that the emphasis was more on a documentation of the process by which you go through the development and determination and evaluation of the models, such that other people could look at that documentation and say, okay, I understand how they did this. This makes sense. You followed good practices, whatever those happen to be, et cetera, as opposed to trying to defend or justify the criteria by which you ultimately decided to determine validation, right? So it seems like it's less of a scientific question and more of a process question. And because of that, the third-party nature of it has to do with the ability of someone who is not necessarily in direct communication with you, who happens to read your papers. Like the last example, I think, is a great one, where if you have a paper, you give enough information so that at least someone can evaluate how the results in that particular paper were arrived at. That is, I think, the sort of thing that we were looking for. And I think the challenge, that was also noted was that since all these projects are in evolution, it is extremely unrealistic to have like a code dump, have someone be able to actually instantiate that model, run it, and determine whether or not it actually runs the way you said it ran. I don't think that's a very reasonable expectation for these projects that are in evolution, but rather that the documentation of the process that goes around the development and the sort of sources and the way you're thinking about how you might ultimately disseminate your quote-unquote finished project, that's kind of what I'm thinking more of. And I'm perfectly willing, as always, to be corrected by other people who know more about this than me.
Gary, can I ask a question? So I get what you're saying. How does that differ from when you write a paper and you submit it for peer review and you get told exactly what the process is and how you arrived at the assumptions and why they're valid and et cetera? Can you distinguish that from just a good paper review where you've got, appendices and all the data and all that?
Well, so the good adjective is a very important one, right? Because I have a high degree of suspicion based on my own experience as both a writer and a reviewer that that doesn't happen. So I would expect maybe a little bit more rigor if on someone who's specifically tasked to do that. And it very well may be that it's information at a pre-publication state, right, where you don't necessarily have fixed results, but you're in the process of developing that, and therefore one of the things that might not be evaluatable is whether or not it produced a particular results set or whatever, but at least that you would have the plan to figure out, that that's actually what it did and what to do if it didn't happen to be that way.
I don't know if this is an appropriate thing for researchers, but of course the FDA has a requirement very similar to that in the design controls process. So if you're trying to design medical devices, you're required to document every step of your thought process, including every revision on the design. And that's an attempt to be able to validate the thinking that went into the design. And then there's subsequently sort of the quality controls process in which you're trying to ensure that the thing you designed is actually the thing that you produced and implanted into the people, right? So it's quite burdensome, however. These things run to hundreds or thousands of pages, and I'm not sure it would be a great idea for that level of documentation in investigator-initiated research.
Yeah, I don't think it's something like that, because I don't think the intent is to be so onerous and but it should have enough flexibility, to account for changes in how you're going to address a particular problem that you're dealing with. But I mean, just thinking in terms of implementation and what this actually would, because we struggle with this, we struggle with this. So I think, some sort of sketch about what that overall process would look like without that, degree of detail, I think would be a useful representation with different tiers that would allow you to dive down based on how far you happen to be along in your particular project.
So, to follow up on that, if you actually go to the poster, there is an example problem on there which basically outlines, what are the things you need to do in order to, at least with the 10 simple rules, to demonstrate that you are satisfying the level of documentation, the level of information that's necessary for possibly somebody to be able to reuse your model. And also in relation to the data standard that was discussed, a couple of things that we identified very early on was actually the second rule, both from when we reached out to the broader research community and within the committee, the second thing we identified was to use appropriate data. And obviously, within each field, there are methods that are there's been consensus at the very least on how to identify the most appropriate data to use. And the documentation process of that is basically what would, sorry, I'm losing my thoughts here. But in general, if you go to the example, it will give you some guidelines in terms of what you need to do. It's definitely not at the level of a regulatory submission process. I would never expect that from a research project because it's in constant evolution.
And I think, just to add to that, that one of the goals for the committee is to get people to think about this and start integrating it into your research. Not so much that we want you to document every decision, but to get you to start thinking about it and think about how we as a community can reach out to others and allow them to believe more in the models that we're developing. And so if you look at the poster that Lalam put together, he's got a very you could say kind of a perfect example where every rule is filled out. But our intent is not so much that you go through and be able to check off every box. Those are guidelines. We don't specify exactly how you fulfill those guidelines, and we did that on purpose because it's going to vary with the model, with your application.
Yeah, and a lot of times what we will find is that naturally as part of your process, you already do those things. It's just the communication process and how you organize it. That's really, a lot of times we find that's what's missing. So, and the idea is that we're trying to find some kind of structure so that when you do present your models, like whether it's to clinicians or other researchers, they have a degree of confidence or understanding of, okay, I understand what they did and I can move forward with this.
So, Herbert.
Yeah, thanks. So I come from the synthetic and systems biology community where these questions are coming up all the time. And for the last, 5 years, we've been trying to sort these problems out. If you talk to the people who build the repositories, the CellML repository and the biomodels repository, they will tell you that almost 100% of the models they get to curate are not reproducible, right? In terms of repeatability, it should be easier. So repeatability is basically taking your code, following your instructions, compiling, pressing the button, we get the same numbers, okay? That should be much easier. Reproducibility is a much, much more difficult problem, and it's been really hard for this community because it's so diverse. But repeatability should be possible. So one thing we do when we build software, one thing I insist that my students do is they'll write down the instructions on how to, and then we sort of get a third evaluator and say, well, you try to build it then. And it almost always fails, 100%. And I have to go through about 5 or 6 iterations with them before, I'm one of the third-party ones as well. And if I get stuck in I usually get stuck in the first 30 seconds. And we go back and back and back. And what happens is, of course, the students are so familiar with the work that they miss out steps which they think are completely obvious. And you could go and visit the lab, of course, like somebody suggested, but we can't all do that. And so the instructions should be very, very clear. So at least the first step, in getting things, in helping with credibility. I mean, credibility is a huge umbrella with lots of things hanging off it. One of them is repeatability. And at minimum, you should have full instructions. And one of the things that the third evaluator can do, and I think that's set up this way, is can they build a source? Can they run the application? Can they get the same results that the original person got? So a long way to go, but I think repeatability would be the first thing to tackle. And it's a culture change, basically. I mean, there's no technical issue. It's a culture change. So it's to instill in students and postdocs, you must be very explicit in what you do. And then once you've got repeatability sorted out, you can start thinking about, well, how can we get reproducibility done? And you can move ahead, and then you fill in all the gaps in the credibility umbrella. So.
Thanks, Robert. That's a great comment.
So I'm not entirely sure we're doing a very good job yet in defining who this credibility plan is, or at least I'm still very confused whether we are trying to make our sort of code credible to other people who are in the same program as us. I can understand code, but they just want to make sure that we actually do all the required steps, or do we actually want to make it credible so that, I think a lot of us are basically working with doctors, right? So do we actually want the doctors to trust what we're doing? Because if the second one, I'm not entirely sure that having another mathematician engineer coming and saying, yeah, actually the code is good, is going to make this doctor say, well, I guess I have to start using it now. I think that we need to have different approaches. I still think that there is a really good sort of advantage of having someone come in and making sure that the process you're following, the code you're producing, the maths you're sort of developing, they're as credible as possible. But this is for the benefit of our own community, so that when I produce something, then someone else can go and say, well, that looks interesting, and actually it has been tested, validated, explored by someone else, so I can collaborate with David and maybe start a collaboration. And that's great. Then we can start having these external reviewers who are people potentially from the MSM community. If we want to sort of make sure that this is something that doctors are going to trust, I'm not sure that someone here is going to make our work a lot more trustworthy to other doctors. So we have to think of different approaches. I still think that there's something else that needs to be done. I still haven't found it myself. I work with doctors all the time. Usually they trust everything you tell them as long as it agrees with their preconceptions of what's going on, which somehow defeats the purpose a little bit. But if that's the second route we want to take, we have to have a different conversation than the one we're having so far.
Yeah, that's a total different job. Yeah, that's communication specialist, that's education, that's as much money as each of our grants individually has put together to do that. That's a huge task, I think.
Actually, I would sort of say that you solve that problem by recognizing the different kinds of models and different kinds of credibility. You know, if you provide, so I'm a physician, right? So if you provide me with a box that has some complicated mathematical formula inside it, I don't understand it, but it tells me which chemotherapy drug to give and it works, I will use that. It won't contribute to my understanding, but I'll use it. It's just a tool. You provide, your model is just it's a software tool. I don't have to understand how it works. If you provide me with a model that changes my conception of the way in which chemotherapeutic agents act on certain types of tumors, now that model, I'm not going to use your software. But your software and your model has changed my understanding, right? And it seems to me that those are two potentially different uses, both of which will appeal immediately to physicians. Physicians are very easy to convince, as long as the bottom line works. A p-value of 0.05 is not good enough, right? It actually has to work, and it has to work in a significant way. And just recognize the difference between tools and things that contribute understanding.
And I think this goes back to, if you look at the 10 rules, is the context of use, right? You want to make it credible within the context of what you're claiming it can do, and so that, you know, you decide what your model is suitable for, who you want to target. Do you want to just predict the right chemotherapy, or do you want to show you want to show the mechanism? So how you decide your model is going to be used, that determines the level of kind of validation and verification that you need to do. John?
I think sometimes we name activities and then we're stuck with the name. This was the Committee for Creditable Practice. It doesn't say it's the Committee on how to accredit models. And so they're two totally different discussions. And so creditable practice involves identifying those things which, if done to varying degrees of documented or correctness, within reasonable cost. And that reasonable cost is a factor, and it might be related to risk. How much am I willing to spend to go to how much detail to document assumptions for a model that has low risk, or at least early on, but that the practice is that the assumptions are documented to some degree of detail. That's all it is. And if you follow the practices, there's an implication, if they're picked well, that the likelihood of having a constructive, useful model goes up. That's all it does. After you have the model, you have a whole different world of how do I accredit the model for its initial use, and then even more difficult, maybe how do I accredit the model for some other use that looks like the original one, but it's not quite. Well, the interface between affordably being able to reuse it may be very much determined by how the creditable practices were implemented when it was created. But if in panels and discussions, we keep trying to mix up the accreditation of models with the development of credible practices, we end up in this endless mess.
Thank you, John. And then we'll take, any comments on that?
It's a really good point.
Succinct.
All right, so last question before we wrap up, and we'll have a chance to discuss these issues more after the second session as well.
Yeah, so I'm coming to multi-scale modeling with a background in genome scale modeling, and you know, when you work with a lot of genome scale models, especially other folks' models, you start to notice which models are really good and which ones really aren't. And after a while, you kind of develop in your own mind, well, I could do this way better. And one thing that was really helpful is there was one of the people who developed these genome scale models developed like this protocol. They published it in Nature Protocols. It was like this 89-step protocol for developing a genome scale model that would actually be useful for other people to use. Like it would actually, people would just put like metabolite abbreviations in there, and you would have to sit there as a human being and try to figure it out because the machine couldn't figure it out, right? You couldn't relate. So there's like all sorts of things that, people who are just trying to study a phenomenon might not think about when they're constructing their models. And this is kind of laid out. This is what you need to do to actually get a really useful model. And that was something that was like, then I could actually say when I published my model, I applied all 89 steps and I automated these parts and all that kind of thing. So it helped raise the level of quality of all these different models that were being published, calling themselves genome scale models. So, obviously multi-scale modeling is kind of different because everybody's doing very, very different things, but for any one group that is developing their models, it seems that being able to document the protocol that you go through to arrive at the model and thinking about that in terms of something you would want to publish so that other people could do something similar might be very useful. And so that was my, I guess, but and that's useful for any one, I guess, particular scale model. Like the thing that keeps me up at night is wondering how I'm going to upscale and downscale different components of the model with each other, and that's why I'm really interested in uncertainty quantification, because they have really good methodologies for combining high fidelity models with low fidelity models, and so I think kind of more communication with those folks would also be helpful.
Thanks for bringing up that example. I think it actually ties in really nicely to one of the efforts that is Anthony here, Anthony Hunt, that one of the members in our committee, Anthony Hunt, has started along with Lalam about trying to document workflows. So if people have examples of workflows that they think demonstrate kind of their good practices, please share them with us, and we're trying to figure out a way to kind of collect these and put them out there so other people can learn from them. So thank you for bringing that point. So,
if I may comment, actually, if you don't mind, if you can email one of us that particular paper, that would be very useful, because as part of our effort, one of our rules is conform to existing standards or guidelines. So within each field, we understand that there are generally guidelines and standards that have already been published, and we want to make sure that people adhere to those so that somebody who's in the genome field, when you publish something, they understand exactly what you did. Now, what we're trying to do is come up with a structure that applies across all scales. But we understand that, you know, if you just try to follow one standard for your domain, it may not always apply. So that's very important. Thank you.
So thank you all for a really stimulating and engaging conversation, and we'll get to continue this some more after the break. Oh, we have...
Hi, this is Ahmet Erdemir, and I just want to make a few announcements. And if you are interested in being a third-party reviewer, you know, please, you know, network with the investigators during the break and talk and see how things may pan out. And also, if you are interested in the committee, we are always looking for active members if you want to get involved, including the investigators there. And I think that's about it for now.
So will we reconvene at 3 o'clock in this room? Enjoy your refreshments.
If I may also comment one thing, to all the investigators, including the ones that just were on the panel, the committee is available as a resource so that if there are specific things that you have questions about developing or credibility plans, just feel free to contact us.
These were Lala Mulugeta and Ahmet Erdemir and Joy Koo. They are all very much involved in the Credible Practice Committee in the Multiscale Modeling Consortium. And I have to say that it was the committee's recommendations that inspired the IMAG program officers to incorporate the language that we did incorporate in the funding opportunity for the model credibility plan. And the model credibility plan, as I describe it to anybody who inquires about it, if they're interested in applying for the funding opportunity, is really the very first start towards model sharing, which is what we started this consortium discussion 10 years ago about in terms of how do we do model sharing. And so the very first step in model sharing is to try to convince someone that your model will be useful for them and that they will like to use it. And I think in one of the last discussion items was what kind of sharing are we talking about? Are we talking about sharing among each other or to the docs in the hospital? And it's really about all of them. And it's really about where you are in the model development. And so I think this is a great discussion to have. And we're really appreciative of the committee to have this portion of our meeting to discuss these plans. It's going to be an evolving process. So we hope to continue this every year. So I'll turn it back to Lalam to continue the session and take it away.
Thanks, Grace. Our next speaker is Dr. Scott Diamond, and he'll be speaking on the credible plan for multi-scale analysis of trauma.
So this is a new project, collaborative project. We're working with hematologists and trauma surgeons at Penn. Also, Talib Sino in the engineering school is helping us with the numerics, and Yannis Kevrekidis at Princeton on some of the multi-scale analysis aspects of the project. So I was told if I push this button, it will advance. And I'm definitely pushing it. The one on the right. Let's try the other right.
No.
So what we're trying to do is patients come into the trauma unit and some bleed far more than you would anticipate based on their injuries. So what the doctors want to know is how, at the earliest possible time, can they identify individuals that are going to bleed And if we can tell them, oh, this individual in 6 hours is going to be a real bleeder, that would be incredibly valuable information. So we take very much an engineering point of view of trying to understand blood as a reactive fluid, but understanding that it's operating in the context of both the systemic cardiovasculature and also traumatized tissue. So these patients are not like healthy individuals. They're really have undergone a very severe trauma. Blood clotting, we know a lot about it. We've been studying it for many, many years. We think of it as a particle aggregation problem, and we also think about it as a polymerization problem. Those are the two fundamental things going on. All occurs under flow. There's some very detailed models, systems biology models of this, and we're working in a community that has developed multi-scale models for coronary thrombosis. We want to take some of that expertise in intravascular clotting and apply it to hemostasis, the ability to make a clot to stop bleeding. It's a very different physics, but the biology is the same. In our multi-scale model, importantly, some of the hardest aspects are describing the platelet, and this is data-driven. We can take a small blood sample and analyze the platelets under hundreds of conditions and basically train a neural network and embed that neural network in every platelet in these fully resolved thrombotic clotting events. And we also have a range of microfluidic tools that we can take blood samples from the patients and run them through these fluidic devices. This is just an example of which we published in a trauma journal. If you reduce the hematocrit, you reduce the ability of platelets to get to the wall, and this is clot buildup with time in this microfluidic device. Very severe defect as you reduce hematocrit. So oftentimes trauma patients are given bags of red blood cells. The clinician's often thinking about hemoglobin, but we're thinking about biorheology and hematocrit. If you take blood from a trauma patient and run it through one of these microfluidic devices, often about half the patients will show a very severe defect in platelet function. We're trying to understand what that defect is. So we have a combination of a patient who's bleeding, who's lost blood, whose cardiovascular hemodynamics are changing, and the chemistry of the blood is changing. This is the problem that we face. Now, these microfluidic devices, we validated them as an experimental tool to give us real-time kinetic data for healthy blood samples, and certainly picks up bleeding disorders. This was hemophilia A, a genotype, very severe performance of phenotype in these microfluidic devices. So from a systems biology point of view, if you read the literature, you can connect a lot of modules and components of the vascular system together, but in reality, this is occurring at many length scales, and this brings us to our sense of the multi-scale aspects of trauma. And let me go through these different pieces because this will lead to our credibility model. So we know a lot about the kinetics of the coagulation cascade. We can actually measure that in the laboratory with plasma samples from patients. We know a lot about platelets. We can measure a patient's responsiveness, the responsiveness of their platelets in the laboratory. We can embed that data into full-scale coagulation models. We've got a number of publications on how to do that and predict clotting with time. Now, there's additional aspects that the endothelium becomes dysfunctional in trauma patients, and you have a lot of edema, and that's a new area that we have to understand. So this is the cellular scale, the vessel scale. This level is very computationally expensive, but ultimately we have to embed these bleeding and clotting events into a traumatized tissue. So we're developing vascular models, but those traumatized tissue beds have to be perfused, and that gets us to the patient scale of how is the heart beating, how much blood is there to pump around, what is the anesthesiologist doing in terms of resuscitation fluids. Ultimately, we want to collect very well annotated data sets for 50 patients, hopefully 50 patients a year, but it's not trivial getting data in real time from trauma patients and extracting that data. I'll talk a little bit about that. So let me just go through I'll skip through this. This is all online. These are some mathematical details. This is the expensive part of the calculation, and I'll come to that in just a minute. So we have three scales. The global scale, we have to choose a model, and there's been a lot of work describing global hemodynamics, control of blood pressure. There's a huge hypertension literature, and these are basically hydraulic circuit models. We don't know so well how certain regulatory control mechanisms work in trauma patients, but ultimately what we want to do is predict blood pressure, heart rate, oxygen, and the concentration of blood factors at the systemic level. That blood is pumped through tissues. We have to choose a model of a vascularized tissue, and there's a number of 1D models of vascular networks where you can control the topology and the pressure dynamics in that. And then finally, the cellular vessel scale of clotting. We ultimately want to predict, can the blood clot and prevent bleeding? So those are things that we want to predict. We're fortunate that we can measure in the trauma patients. We get real-time readouts pushed to the to Epic, basically, heart rate, blood pressure, oxygen. There's a lot of blood chemistry that's measured on a fairly frequent basis. The tissue level is particularly difficult because that's something we really can't get our hands on or look at or measure. We're working with our collaborators in a mouse injury model. That's both a hemorrhagic shock model of hypovolemia, but also trauma. Those things two put together create this a very difficult mouse model, but that at least allows us to get some data at the tissue level. Patient data, we only have some metrics that we can work with. Estimates of blood loss, not particularly precise, and then blood product usage. That is a very precise metric, a way of keeping score of how much blood is coming out of the patient is balanced by how much blood products they're putting back into the patient. At the single vessel level of hemostasis, we're very fortunate in this situation that we can get blood samples with time, so we can do lots of functional phenotyping on that blood, tags, platelet mapping, microfluidic tests. We learn, and we since save the plasma samples offline, we can do proteomics, metabolomics, so we get a lot of data linked to that blood sample in that patient. But we have the challenge of interfacing these scales, and this is like many of the groups, and it was mentioned earlier, some of the models you can only solve for a certain period of time, maybe 20 minutes. You know, there's a computational cost, but we really want to predict the patient trajectory over a long period of time, maybe a 24-hour clinical experience. So that's where, you know, techniques like course projective integration may become very important to us. We're already working with our end users. We're working with trauma surgeons. We're working with medical informaticists here at our university. We meet with them often, and we see them often. It's all around the patient. So we're kind of embedding the end user up front because we want to make things relevant for them. And I'll finish with this one issue. The data from the patient is sitting in Epic. I refer to that as a data jail. It's not trivial extracting that data, but we have to marry that data with our research data into a database, and this is proving fairly challenging. All that data extracted from from Epic is HIPAA protected, and that probably won't see the light of day, but will be made available to researchers. Okay, thank you for your attention.
Good presentation. Our next presenter will be
Great, thanks. I'm going to talk today about our model and some of our work in cystic fibrosis. The main idea with this model, or our main goal, is to create a systems model that can kind of bridge the in vitro studies we do of therapeutic response to in vivo outputs of therapeutic response. And we're working specifically in cystic fibrosis. And cystic fibrosis is pretty much an ion transport defect caused by a single gene. And it results in some problems at different epithelial surfaces in the body, including the lungs. And what I've shown here is kind of a section of the airway wall. In a normal airway, these ion channels help to maintain hydration of the airway surface liquid layer, and that's very important because mucociliary clearance, which exists in the lung to kind of clear out particulates and pathogens that we inhale, depends on that hydration. That little thin layer of mucus up here will carry that stuff away as the cilia beat and kind of drive it like a conveyor belt up and out of the lungs. What happens in CF, though, is one of these ion channels is actually missing, and that results in mucus just kind of accumulating on the surface. It becomes dehydrated. Homeostasis is lost in the ASL, and kind of defeats mucociliary clearance. So things we're going to be interested in putting into the model are things like mucus clearance rates, airway absorption rates, electrophysiology to really account for some of those ion channel defects. We've been successful in developing a primary organ-level model to measure mucociliary clearance and airway surface liquid absorption using some imaging methods that we have. We're working with cell culture models, which are really common in CF because they don't really have a well-developed or fully available animal model. We're trying to make the transition from using bronchial epithelial cells, which we get from lung transplants, to using nasal cell models, which we can sample from patients non-invasively, so we can make other measurements and compare the physiology we see in the cultures with the physiology we see in their lungs. And part of our U, which is, and this is our first year, is doing some studies of lung physiology along with measures of these nasal cell cultures from the patients, and also tracking clinical trends in some of the CF patients, and then trying to tie all this information together. And I've just shown kind of a smattering of the measurements we're making using nasal cells. So we get nasal cells from all the patients enrolled in this. There's patients, single mutation carriers, and also healthy controls. We culture those cells. We measure some things in the lab about electrophysiology. We then bring the patients in, and we have kind of a whole set of different measurements we're making of lung physiology, some of which are kind of unique to our center, the small molecule absorption, mucociliary clearance, and nuclear imaging techniques that we've worked on a lot. And then we're also going to follow the CF cohort of these patients in terms of pulmonary function trends, clinical trends in general. Try to tie all this together with a model. And like I said, the ultimate goal is to be able to go in, take nasal cells from a patient, grow them out, make some measurements in the cells, maybe put some therapies on the cells, put that information into the model, and then determine what therapies are going to be effective for that patient, or use a host of different cultures that just kind of make some judgments as to what therapies we should be developing and doing clinical studies on. Right now, we are working in these models all separately. They're all underway to some extent. And the goal is eventually to take our cell-level models, combine them into this larger organ-level model, and make one kind of comprehensive model that'll take us from the nasal cell measurements down to a prediction of organ-level response, and then hopefully someday to clinical outcome prediction. So this is where we're at. We're recruiting this many subjects as part of the U, getting data on this many subjects. We've got, I think, 9 subjects enrolled so far. In terms of our plans for validating the model, we started off, at least internally, with a very simple plan, which was we're going to take some number of subjects, use that information to inform the model, and take the second half of the group and use that to at least initially validate the model within our group. We've also discussed some of these cross-validation schemes where we would take the whole group and pull groups out of there, or data from different patients out of there, and kind of do a cross-validation within the group. In terms of a third-party evaluation team, we were strained to kind of think of who would be the one that would go along and evaluate this, because I think we've put together kind of a unique group of people, and I'm not sure there's another group that's exactly like it. We might be looking at something where we'd have to provide a data set in the model, and I'm just not sure anyone out there would be able to provide fresh data for this model exactly using the methods we have. But these are just some of the things. I'm not going to read through them individually that would be helpful in terms of a third-party evaluation team, and we're certainly interested, if someone has an interest, even if it's not in all these areas, if it's in a few of these areas, in talking to you about being a third-party evaluator. In terms of where we are for having something to evaluate, we've got an organ scale model that we published on, that we'd worked on really ahead of the U, that we certainly could let someone else look at, would like someone else to look at this point. We are working on the cell scale models. We've got kind of a basic thin film model together. We're working on what we consider to be our oozing chamber model, which is more of an electrophysiological model. And we certainly could be able to make those available relatively soon for evaluation. The integrated model is probably a ways off. We have to get both of the subsets of the models, the organ level stuff and the cell level stuff working, and then integrated before we have that to evaluate. Challenges, small population. We're always working with small numbers. There's only about 30,000 people in the U.S. who have cystic fibrosis. We're never going to have hundreds of patients to look at unless we're willing to stay at this for a long time. Some of the methods we use for gathering data, especially the imaging methods, are pretty unique to our center. The cell-based methods are not. They're unique to many of the centers, especially those that work in CF. And there are consortiums out there through the CF Foundation that may be helpful for us because they're working with cell models and they've already standardized some of the techniques that are included in what we're doing. This is just a little bit of information on how we were broken down as a group. I wanted to make sure our contact information was in there. I'm the guy who does most of the imaging. Bob Parker was here earlier today, does most of the modeling, and Carol Bertrand does our cell culture models and our electrophysiology. And just to kind of end with some of the points that we were asked to hit, these are just some of the things we see as, how we fit into the structure about model practice. This mostly is written in Python right now. Some of the earlier models are written in MATLAB, and we're trying to convert most of them over. It's just a little easier for us to work with Python. And just kind of end with who we think this would be interesting for. I think our ultimate goal is to put this in the hands of people who have therapies, prospective therapies for CF, so that they can kind of make some better decisions about what therapies they develop by just doing the in vitro studies rather than having to do 6-month clinical trials of each therapy in the pipeline to find the best. And it'd be great someday if we could put this in the hands of CF physicians who could use the model to kind of prescribe personalized therapies for these patients, figuring out what medicines are going to work for them without having to go through the normal trials that they do with patients. And I will stop there. Thanks very much.
Will be Dr. Silvia Blemker. I hope I said that right. All right, we're happy to go through this.
Hi, everybody. Glad to be back up here. I'm excited to tell you a little more about our project and specifically how we're validating and verifying our model and making use of some of the credibility requirement suggestions proposed by the working group. Again, I'm Silvia Blemker, and my co-PI in this project is Shayn Peirce-Cottler, who's also in the room. So just a reminder what the goal of this grant is, really kind of rule one, which is what's the purpose and what's the context and who's the audience, is our goal is to develop multi-scale models that predict disease progression in Duchenne muscular dystrophy. So we want to create models that replicate what actually happens in boys when they're first born with a genetic mutation that leads to lack of dystrophin, how over time that leads to muscle degradation and wasting, and from kind of early stages to late stages. And we're interested in this whole scale from modeling the loads to the tissue scale and then modeling the changes in the cellular environment. Ultimately, we want to use this to test new potential treatments for DMD. So a little bit, a reminder about our framework, because this relates to how we're going to validate. So we have one scale, which is finite element simulations of a whole muscle. We're using FEBio for these simulations. And those are going to provide the macroscale strains that are going to be, or deformations that are going to be input into our micromechanical simulations, which are smaller scale, still finite element models, still done in FEBio, but a scale down that represents fibers and connective tissue here. And then those provide the mechanical signals that are inputted into the agent-based simulations that also incorporate several rules that dictate the behaviors of the cells within the muscle, incorporating the mechanical environment, but also the biochemical environment. And that predicts over a given time frame changes in the muscle over time, remodeling, degeneration, regeneration, depending on the nature of the loading. And then that will update our micromechanical model, and which will then be updating the macroscale model. So there's a lot going on here. But ultimately, the goal is to predict the remodeling of the muscle at both the small scale and the big scale, and we're doing observations of muscle wasting at both of these scales as part of this grant as well to validate the modeling predictions. So the key components, basically, verification of the modeling code, both the good thing is that we're leveraging existing code for the basic components, both FE-Bio for finite element analysis and RePast for the agent-based simulations, and those have their own sets of kind of already prescribed good verification tests. Obviously, so our verification is going to be focusing on the interface and how these things talk to each other, and we have we're developing some test cases to start with to make sure that that the code is verified. And then validation is a big kind of a lot of our project is actually focused on validation, sensitivity analysis, documentation of model limitations, and then the last bit that maybe is a little different from the rest, speaking to the uniqueness, is we also really need to evaluate the credibility on the clinical and biological side, i.e. are our predictions and simulations consistent with what is observed clinically in this population? Because if it's not, then the whole thing's kind of a waste of time, because the people who we really want to understand our results aren't going to believe them. So this is a very busy slide showing all the aspects of how we're integrating data into the model. So we have, in the whole muscle simulations, we have a number of data sources, MRI and dynamic MRI, force measurements, that are all going to go into feeding into the model. And then we have a number of measurements that are going to validate the crossover between the whole muscle simulations and micromechanical simulations. So we've decided to validate internally each component, and then validate how the components are talking to each other. And so I don't want to go into all the details, but I'm happy to talk about them with you. But we think it's important to do those separately, because you can imagine errors to be propagating if you only validate how things kind of come together. As far as our timeline, we've outlined a lot of our time is spent on validating the beginning validating individual components, which we've done a lot of that already in our previous work. So much of our focus is the validation of these couplings, A to B, B to A to B, all these various coupling. And then we're also, the next phase, so that first phase is developing models that are consistent with observations. The second phase is now testing the predictive capacity of the model, so validating that piece. So developing simulations of potential drug treatments, which we're going to test out in the lab with preclinical studies with the mouse model. So this is now kind of the tested of the predictive capacity, really using the models to guide the experiments, to guide the hypotheses, and see if those hypotheses are coming through. And then the last one is I described before, this humanizing of the model. So the way we're validating that is by we're tracking a cohort of boys with muscular dystrophy that are attending our muscular dystrophy clinic at UVA. We're tracking them over a course of the whole grant and developing models of each boy and seeing and testing our ability to predict for each boy how their muscles are degenerating. It is a very inhomogeneous disease in many ways across muscles and across boys. So we think that will give us the opportunity to see if we're at the point with the model that we can actually give insight into that. So a little bit of our progress. So we've been focused on the validation and sensitivity, so comparing predictions for here the agent-based model with experimental measurements of cell counts after injury. So we're at that phase. This is really the calibration phase, our rules parameters. This is an example of a sensitivity study that we've done to look at the sensitivity of the model prediction to here is the extent of damage imposed on the muscle. And then the last one here is maybe a little bit unique, is now testing the model's ability to predict perturbations to the system that have been measured in the literature. So things like the top one is things like what happens to injury, regeneration following injury when you deplete fibroblasts. And so these are what's happened to each of these cells, and the red means decreased regeneration, blue means increased degeneration. And the top triangle is what's observed in the literature, and the bottom triangle is what the model predicted. So this is a little bit more of a, it's more of a qualitative comparison overall. Is our model predicting the observations in the literature, what's generally found? But we think it's a really important one, again, to speak to the sort of biological credibility of our model. And the other nice thing is that we kind of have these uncertain things, these hashtag ones where nobody's done that experiment in the literature. And by doing this has actually helped us identify what experiments we may want to do in our, what measurements we may want to make in the lab. So a few discussion points for challenges and opportunities. The complexity of our models does present a challenge for the robust uncertainty quantification and sensitivity analysis because we're crossing all these scales. The extent of the model really makes it hard to fully do a full uncertainty quantification, but we're really excited to be part of the community and learn the latest techniques that we could potentially incorporate into our approach. The other thing is this combining deterministic and stochastic behaviors. Finite element modeling is implicitly deterministic. The agent-based modeling is stochastic. We have a lot of stochastic rules. So how to put those together and do appropriate validation is tricky. And then the last one I think I alluded to before is numerical validation is all great, and it makes it so that modeling journals will like our papers, but which is important to us. But we also want to have an impact on the broader community of clinicians and muscle physiologists. So, you know, something could be numerically valid, like we've validated in every way, but maybe the predictions are clinically irrelevant, or vice versa. So really struggling that balance is what we're interested in. So a third-party evaluator, we have on the clinical side, we have a close clinical collaborator who's really serving that purpose for us. But then we are looking for two evaluators from the MSM community, ideally one with a lot of biomechanical modeling expertise and another one with agent-based modeling expertise, or people that have both. But if anybody's interested in our project and getting involved, please do come talk to Shayn and I after the session. And we're also looking for a postdoc. I forgot to mention this before. So any graduating PhDs in the room who are looking for a job, we're hiring a postdoc now, and anybody from this community would be a great fit for our position. Thank you.
Great presentation. Our next speaker is Dr. Bruce Lee, and he will be speaking on the credibility plan for virtual obesity prevention model.
So I'll be talking about our VPOP labs, which stands for Virtual Populations for Obesity Prevention and Control. These are basically computational multi-scale models for obesity solutions. So very briefly, the intended audience really are any types of decision makers within the community or city that is interested in obesity prevention and control. So these essentially are what we're developing, our virtual cities. So representations of all the people, all the food sources, schools, households, workplaces, and physical activity locations that are relevant within a particular city. And these can be used to determine the factors and relationships that may be leading to obesity, help forecast different future scenarios and impacts. So for instance, if you have a city and what happens in 10 years, 20 years with the same type of conditions or different conditions, help design and plan obesity-related policies and intervention, help assess the potential impact of these policies and interventions, including their secondary and tertiary effects and unintended consequences, and also help guide and prioritize data collection that might be relevant. So essentially for this project, we're focusing on developing virtual representations of two cities. One is Baltimore, and the other one is New York City. This project has and will continue to heavily involve different stakeholders within each of these cities. So what we've done is we've formed policy working groups for both Baltimore and New York City. So in each case, we have city council members, city health department members, representatives from the public schools, family, recreation parks, wholesalers, retailers. Essentially, we went down the list of people who would be relevant to obesity or relevant to the potential causes or solutions to obesity. So here's pictures of some of our members of our stakeholder working group. Also with New York City, we were initially engaging the Department of Health, and progressively they'll bring aboard other departments like Department of Education, Department of Urban Planning, all those different areas. So this is a highly iterative process, in which we're actively involving them in the design of the model, as well as the types of interventions that they're interested in testing, as well as the validation. So they can provide both face validity, but they can also help with criterion and convergence and divergence validation, because there's extensive data sets. So for instance, New York City has extensive data sets that we can use in part for population and calibration, as well as validation. The other things that are occurring is we're pairing these development of these models with existing continuing concurrent studies. So for instance, in the city of Baltimore, our center also has an ongoing multi-level community intervention study, which essentially is looking at the combinations of different types of obesity prevention and control interventions. So these range from educational programs to social media campaigns to interventions at the store owner level to try to improve their stocking patterns, to recreation center interventions, to policy level types of interventions. So we have these ongoing trials and intervention studies that have been going on for about 5 years already, so that we have a head start and have data already produced from these studies. But these are ongoing, so we can also adjust the protocols accordingly to generate the data that's needed to validate the models. So this is the timeline and milestones for our model credibility plan / validation. So essentially what we're doing is we're building these multiple scales in different layers. So we don't, of course, throw in all the scales and layers all at once. So the different scales here are basically the genetic scale. So each of the agents within this model has a genetic scale within them, and then they also have a physiologic model within themselves as well. Then each of the agents then have individual behaviors in terms of getting up in the morning, moving around to different locations, eating and exercising, etc. And then the fourth scale is the group or social network scale, in which we connect different agents by different types of social influences and social networks. And the next scale right above that is the physical built environment scale, where we're representing all the different stores and recreation area locations, or physical activity area locations, as well as households and workplaces. And then the societal scale, where we're representing larger forces like economic forces, policies, larger cultural forces, and those things. The plan is to initially build and validate scales 3 and 5, and then comparing what we're finding with measured actual daily activity data. So what we essentially have done, and we've already completed this part, where we've put in all the different agents, put in their decision rules and their algorithms, and have them move around. And then they complete different types of daily activities. And then we measure the frequency of these different activities. For instance, how often will they end up going to the corner store? So we have, for instance, a gravity model, which determines their likelihood of going to different corner stores or different grocery locations. And then what we've done is compared those outputs or those measurements with actual daily activity data that we're getting from the literature, from available databases, as well as our ongoing community interventions. So for instance, we found that the daily activity of the agents matches reasonably well with the community intervention study that we're doing, the BHCK, which stands for Baltimore Healthy Communities for Kids, that ongoing community intervention study. We found that those matched up pretty well. So that's one example of matching the activity data with the generated model data. The second step is then determine how well the combined scales 3, and that's apologies, that's supposed to be 5, and 2 model can then replicate historic BMI changes. So here what we're doing is we're taking that initial model, which has the individuals and the environment scales, and then layering in within each of the agents the physiologic model. So we've already done that. And then we then can measure and track the BMI changes over time, and we have extensive historical data on the geospatial changes of BMI. The next step is then adding scale 4, which is the social networks, and then comparing that alone with available data on social connections, and then the combined model 2 to 5, looking at the combined ability to replicate both the daily activity data and the BMI changes. Then part 2 of this is with years 3 through 5. Then we layer on scale 6, but before we're doing that, we see how well the societal scale model then replicates what actually happened in these cities in Baltimore, and then we also have extensive Pennsylvania data, and then New York City. And then finally, then we validate scale 1, which is a genetic scale. We have an ongoing large study as part of our center that collects data on 37 counties in Pennsylvania, and that includes extensive collection of epigenetic data. So then that can be used to validate scale 1. And then, of course, then we combine everything, and then we try to see how well our model can predict the results of different types of interventions that have been tried in the past. So I mentioned the progress. This is what we've completed in terms of the first 2 steps of validation. I call out an example of how we've done the second step, which is replicating historical overweight and obesity prevalence in Baltimore City, and then replicate the daily average of caloric intake of different types of items, for instance, like SSB or sugar-sweetened beverage consumption. So here are some of the challenges and opportunities. So one is these different scales have very, very different types of validation data and associated limitations. So you can imagine the type of data that's available for societal forces is quite different from the type of data that's available for individual behaviors. You're talking about differences in granularity. You're talking about differences in data gaps and things like that. Each of these data sets have their strengths and weaknesses. So the opportunity here is really to help identify and better characterize these gaps. There's cross-learning opportunities, so folks who are steeped in data at the societal or, say, environmental level might be able to learn from how data is being collected at the physiologic level and vice versa. And we can help identify new ways of harmonizing data. Second challenge is validation data comes from different samples, locations, and time frames. So as I mentioned, we are trying to stick as much to data that's native to these locations, like Baltimore and New York City, as much as possible. But we have to use data from other locations, such as Pennsylvania. We're using some national data like NHANES data and things like that. So of course, one of the opportunities to really better understand this variability and robustness, and this can potentially inform data collection in the future, like what are the weaknesses of NHANES, for instance. Third challenge is model descriptions, presentations, visualizations must be, certainly in the case of this project, be very digestible by a wide range of decision makers, including the general public. So most of the people that we are interacting with have never done any modeling naturally, and may not be experienced in modeling lingo or jargon or particular scientific jargon or lingo. So we have to make everything very understandable by a wide range of decision makers. So of course, each of the different decision makers will have their own specific areas, but we have to do that. And so this is a real opportunity to engage decision makers and improve translation. And also we'll be testing some policies and interventions that have not been tried yet in the past. So there's going to be pieces of data that's available for some of these policies and interventions, but they haven't been unveiled in this whole-scale manner. So this is an opportunity to really help shape and design these and help develop these policies and interventions in concert. So the uniqueness of the model credibility plan, we're involving multiple stakeholders very actively. We're developing new translation and communications approaches to new sets of stakeholders. So there are a lot of people who have never had any experience with modeling here. These are new approaches to obesity prevention and control. We're pairing the model development with all these other different types of studies that I mentioned. And this is really an iterative engagement process. So I think I showed this slide a couple days ago, but essentially this is an iterative process. So the stakeholder working group is actively involved in each of these steps, and we're developing the model in a cyclical process where we're continuing to inform the stakeholders, getting more data, and then updating the model. So they're quite heavily involved. So again, this is our system science core team, just to give them props, because they've been doing a lot of the work behind this. And I guess we're going to be saving questions to the end, right? Okay, thank you.
Thank you very much for the excellent talk. Are you? Oh, okay. All right.
Good afternoon, everyone. Thanks for the opportunity, and appreciate you staying that long. So our project is multi-scale modeling of blood flow and platelet-mediated thrombosis. And few collaborators are here, Joanne from Applied Math, and yesterday Dr. Slepian. So just describing the model, what you see here is a model that departs from traditional continuum methods. This is a particle-based model that interface spatial scales from nanometers to millimeters and temporal scales from picoseconds to milliseconds. And basically to bridge the gap between what happens in hemodynamics and initiation of thrombosis that happens due to molecular processes. So you see here blood flowing in a stenosed blood vessel. And already here you have a scale exchange where we go to fine granular infrastructure of a platelet that is modeled by coarse-grain molecular dynamics and the internal parts with molecular dynamics. And each such platelet in itself with bilayer membrane, cytoskeleton, and cytoplasm can be built up currently close to 500,000 particles. And then we also model the activation of platelets due to the loading history, which happens, kind of dose-time response of platelet activation that induces the generation of pseudopods and creation of aggregates. At that stage, we have to coarse-grain this very fine-grained model and reintroduce it to the blood flow stream, where it's going to affect the hemodynamics. So the hemodynamics are modeled by dissipative particle dynamics, and then there's the interface to the platelets and the activation process. Now, basically, the model is able to predict the changes in platelet morphology upon this shear-mediated platelet activation and biomechanical transduction processes, and also interaction with clotting factors, aggregation, and eventually adhesion to the blood vessel and thrombus formation, or it could be also in medical devices. So the audience or stakeholders that could use this model, clinicians and scientists, to predict the initiation of arterial thrombosis under circulation conditions, unlike the traditional methods that usually test this under static conditions. And one of the things that we found out through the predictions of the model, that we can develop new approaches for targeting mechanotransduction pathways for developing pharmacological management of thrombosis. And eventually this may lead also to the use by cardiovascular device engineers. Now, so regarding our model credibility plan, the verification and validation strategy, which of course also depends on the context of the using the model. So validation is done through geometric, rheological, and material properties using in vitro experiments, and also the data that is out there in the literature, not too much for flowing platelets. Shear-mediated platelet shape change, we're using our own hemodynamic shearing device and also scanning electron microscopy. We also monitor the flow-mediated platelet flipping and aggregation while it's flowing through the blood vessel, and then adhesion into the microchannels. We do that with video microscopy, and we already starting testing antiplatelet agent-induced membrane fluidity to increase the platelet resistance to shear activation. In that, we are also, of course, concerned with uncertainty quantification and parameter sensitivity analysis, and we have quite a few of those. Now, this is the general timeline of the project. So it started already with funding via an R21 from NHLBI and also previous year one quantum awards. So we kind of starting to mature that with this new year one that started 8 months ago. Now, regarding what we achieved right here, now, so in the verification of the model, we want to test the ability of the model to use the actual mechanical properties of platelets. So the more traditional method would be micropipetting. So we basically numerically mimicked micropipetting in this numerical experiments and compared it to data published in literature, but Hager and assimilation got a very good agreement, and we have both the young modulus and shear modulus of the membrane, the plasma viscosity, which is not an easy fit for particle-based models, and also the cytoplasm viscosity using all type of Morse potential and modified Morse potential for those in the know. Now, Dr. Slepian yesterday also showed this slide. We developed this electrodeformation method in order to measure the mechanical properties of platelets without touching them, because platelets are very capricious creatures. You use atomic force microscopy, touching them, you may activate them. So here we stretch them by electrical currents and electromagnetic stream, and then we can measure the mechanical properties of the whole ultrastructure of the platelets. Now, the actual numerical experiment is done under here, in this case, pure wet shear flow generating 45 dynes per centimeter square, pathological type of shear stress. So the platelet is flowing and flipping, and we monitor stress distribution upon the platelet membrane, which eventually will push the platelet beyond the threshold of activation. This is a kind of dose time response or stress accumulation over time. This is transduced into the cytoskeleton, the way you see here, and when it crosses the threshold, the model will predict activation. Now, here you see also video microscopy of the platelets flowing and flipping. You can see right here, and it does that according to a certain prescribed frequency. So we measure that in this micro channel with this video microscopy and get very good agreement. Now, what you can see here is the actual process of platelet activation with the extrusion of filopods that eventually will lead to aggregation and also attachment to the vascular wall. And for that, we built a very big database in which we have different morphologies, and we interrogate this parameter database according to the different parameters that you see here in order to tell, and not exactly tell because it's a predictive model, for the model how to extrude the pseudopods and the number of the pseudopods that should be extruded. Now we started doing the aggregation of the platelets, and here you see the experimental validation and what the model will eventually be able to predict. And this is a huge number of parameters that are involved in all the scales. You see here the key experimental parameters, the key independent model parameters, and the adjustable model parameters, and how we plan to do that. Of course, I won't go through all the details. So the uniqueness of our model, it utilizes in-house equipment, hemodynamic shear device, DIC microscopy, microchannel setup, identifying, quantifying the dominant sources of uncertainties in this truly multi-scale modeling. This is, of course, as complex as it is, it's a reductionist model with certain assumptions and not expressing the true physiology, but the real test will be its ability to predict the computing uncertainties, the coarse graining process, minimizing the global uncertainties, stress testing of the high-performance computing resources to see that we're doing the simulations correctly. The third-party evaluation requirements require knowledge of open code labs that we use for the DPD and molecular dynamics simulations, familiarity with both molecular dynamics and dissipative particle dynamics theory, and familiarity with basics of platelet activation, aggregation, and adhesion, also availability of HPC resources, and I could discuss it later in the panel discussion. Thank you.
Thank you very much, and now we'd like to invite all the speakers to come to the stage. To kick things off, I just want to bring up a question that one of the audience members actually brought up, which seems to be a reoccurring theme even from the previous panel discussion, and that is the third-party evaluation. And the question that was raised was, what qualities are needed in a third-party evaluator? How expert should they be in the specific model application domain? These are important questions. And I think Jennifer Linderman, she actually approached me about this earlier. And when it comes to the third-party evaluation, in my past experience, while I was still working at NASA, where we did do various degrees of third-party evaluation, what I've learned in that process, if it's helpful at all, is you shouldn't think of it as something that is one person. Definitely not. It has to be multiple people, pretty much every single time. And you don't have to have all of them test your model. That's the other thing what I discovered. You can have, let's say, if it's a model that's intended to be in the clinic, you can have one expert who has the expertise to run your model, who knows the knowledge base of HPC, for example, and you can have an MD who can actually look at the results and really interpret the results. That's another way of doing it. And then the other thing I learned also is that depending on what spectrum the application of that model is supposed to be. For hypothesis testing versus clinical application, the granularity or the degree to which you're going to have that, the rigor, should I say, you're going to have that third-party reviewed, is going to vary. You know, for something that you intend to put in the clinic, I'm just throwing a number out here, so don't quote me on this. It could be a process that takes a month long, where it's much more rigorous. If it's something that's hypothesis-driven, it could be a 3-day meeting for very key elements of the model and simulation and the data and whatever else that you can have them review. And you should have a documentation process also which says what did you do exactly and what were the conclusions that were drawn from that review process. Now this has been my experience, and I just wanted to share that and also to answer the question, at least from my perspective. So with that, I would like to pass it over to you guys if you have any more thoughts or comments about that.
certainly in our case, it's important that the end users of the information or the model be involved, because many times there are subtleties or perspectives or things like that that if we're not capturing the model, it's not going to be useful. And so there's a difference between quote-unquote scientific validation and policy active validation. So you can do something very well scientifically, and it can be interesting, it can generate some interesting scientific insights, but it may not be actionable from a policy standpoint. So certainly including those folks is important, at least in what we're doing.
I would think that I believe that the true utility of a model is that if it could also be used under different scenarios by other users, but of course to establish the validity first, and if you're dealing with methods that are not widely used, because most of the modeling that is done in that domain or in our specific case based on finite elements of volumes, well, we depart from that, and then a few here, like George Kaniadakis and Mark Aldo, doing the same. So it's a relatively small community in the biomedical bioengineering sciences, although molecular dynamics is very big in biology, but not under flowing conditions like we're doing here. But regardless, while it will be pretty challenging to create a kind of a benchmark that others could test to validate that, and the actual validation that involves both highly demanding numerical simulations and also experimental, very specialized techniques, then I think that the truth is maybe through peer-reviewed publications, but once this gets out there, then people could definitely use the codes that we are developing, because of course we keep very strong documentation there, and we could guide the people how to use it for different applications. Regarding what's the actual utility that in that? So one of the things that I mentioned earlier, that it was born out of the simulations themselves. We've noticed that when we modify parameters in the model regarding the stiffness or flexibility of the bilayer membrane platelets, then we make them less or more resistant to shear that activates them. And this actually led us to look at new therapeutical approaches, because all the traditional approaches look at biochemical pathways rather than these mechanotransductive pathways. And in that, we have many ideas, and this is, relates to Dr. Slepian's presentation from yesterday, because we are motivated because most of the anti-platelets, anti-thrombotic drugs out there are not efficient under real pathological flow conditions. So new ways should be tested, and in that, modeling kind of predicted that and led us to this pathway. So there is a utility to that.
One other thing I was curious, your thoughts on, since these UO1s are 5-year grants, at the beginning, you know, it's very much code development and preliminary prototypes. At the end, theoretically, it's much more of a package. Do you imagine that the third-party evaluators should change then, according to where you are at the stage? Like the evaluator at the end would be more of an end user, at the beginning would be more of a kind of fellow modeler who has the deep technical expertise.
That's a question directly.
Yeah, what do you think? Have other people done that, or what's your take on that?
My take on it is, my personal experience has been, you need to think about, you should have at least somebody that remains fairly consistent in the continuum. Like, let's say if you have 3 different reviews, let's just take that for an example. In the first review, you had one set of reviewers. In the second, you may have, let's say, half of them present, and the other one, you switch them out. In the third review, you may have the others that were in the first one, but not in the second. So you can always mix and match based on the needs. That's the way I've always approached it.
I think that also like addresses the importance of how the audience may evolve during the modeling and simulation life cycle and how we should adapt to that.
Yes. But I think you touched on something that's very important. I found it to be a severe mistake I've made to leave the independent review to the end. You should have a continuous review process. You should have milestones, and you should integrate the independent reviewers in a partial way so that you know you're on the right track. And the other thing is that it doesn't become an onerous task at the end to actually review it. Any other panel members comments?
I just wanted to add to Lalam's comments, and one of the reasons we are actually trying to be proactive with these credibility plans and, with the, committees. Now, we know that, we always need help, and it's better to get the help like at the beginning or help the inputs like at the beginning, because when you do your work and when you write your publications and then you find your model is not necessarily that credible, then that's a wasted effort. So maybe we can increase the efficiency that way.
I guess I want to add another benefit of getting third-party evaluators involved early. The actual modeling process itself is very informational, so it can help people better understand relationships, data gaps, and those things. And so we've had great opportunities where decision makers, they're understanding the model along the way, and they actually have taken us in different directions that we didn't anticipate going in a positive sense. So.
That's a fantastic point.
And part of it goes to Emmett's question about the audience and things, and I'm going to target the trauma blood flow thing. But one way to go is see what happens. Another way to go is that you have a vision that today, if a patient comes in with a partial amputation and a gaping chest wound, what happens? What happens when they roll through the emergency room door? And in your vision, how will that be different in 10 years if this model becomes significant? What's going to change? And there's 2 reasons for doing that. One is so that you have some benchmarks for expectations along the way, and the other is you may do things with the model to make it usable for that purpose that have nothing to do with the model itself.
So we view a patient as a high-dimensional trajectory. We want to understand that trajectory. And the computations, the modeling, could actually take many days to run. But if we can quickly look at a patient coming through the front door, take a blood sample, measure those 6 things quickly and say, oh, that looks like those 30 simulations that we ran last year, we're going to put that patient in the bin because we know what that high-dimensional trajectory is. That's an endpoint for us. That's where we would like to be. And you don't have to run the model for that patient because we've already run the model a lot. And we would love to be in that position. That would be our goal, to have 10 bins and a way to wrap it, those 10 bins being very well explored computationally last year, but having a way of putting that new patient and the next patient into bin number 3 because they've met these things that we discovered along the way of developing the model. But unfortunately in trauma, they don't want to wait 5 minutes for a data point. So I can't run these multi-scale models for that patient in real time, but I can run some diagnostics.
But using the model to pick up other markers that
Exactly.
may be observable.
So biomarker prioritization is an outcome. It is, you could perhaps go to an end user and say these 7 biomarkers are really important for you, and then you could do the study and find out.
And that's, I'll be really disappointed in 20 years when I go to the hospital if I don't pass through the full body scanner on the way in the door. And in terms of speeding up the activity of having this kind of data about the patient coming in the door.
In all this discussion about evaluation, I'm not sure if I fully understand, maybe it was specified, and maybe it's a question to Grace, partly, is how is this evaluation process going to be documented? When is it going to be shared with at least the consortium versus a larger community? In what form? Is that going to be, we have to wait for the paper for it to be coming out? Is that going to be an annual report? Is that a continuous wiki update? What is the modality with which the rest of us can learn how the evaluations are ongoing?
You're going to get a request to review a manuscript. That's going to be the first sign. I still think, this is an opinion, that we still as scientists have an obligation to publish in peer-reviewed journals, and that's usually a very important milestone for our trainees and for documenting the research archivally. And depositing models in repositories, I think, is linked to that publication event. I think that's very important. You know, there's deeper issues that you've asked about, but I still feel peer-reviewed publications and depositing models is a very valuable first step.
I just want to be clear that that's not the same thing as evaluation we're talking about, right?
I think Grace was going to take that question.
Say something.
I don't have an exact answer right now for you, Raj, but I think this whole thing is a process. I mean, if we're going to follow the usual grant route, these U01s will have science officers that will look at the model credibility plans along with the progress and everything would be internal. But the whole purpose of having this session today is to make it part of the consortium and to get the consortium's feedback on how these credibility plans are set up and to give any advice to the investigators on how to do it better. Like I said in the intro, I thought maybe we could do this each year at this annual meeting. I'm not sure in what form. You know, as we get more and more grantees, it may take up the whole entire meeting. So it may be that, you know, we work it out with the steering committee as well in terms of where do we want to use this forum for this activity. But certainly it should be public as needed.
Can I add? I just wanted to add something. So Peter Hunter at Auckland, the ABI, he's starting a journal. Have you heard about this? I think it's just called Physiome Journal or Modeling. I forget what it's called. But the idea of it is to pair with Scott's point about peer review. When we publish models, we write a paper about it, and it's focused on maybe the kind of theory of the methods and the results. Maybe we, and that's peer reviewed. And maybe we post our model code, but it's not like anybody, you know, that's sort of like, oh, here we share it. He's developing this journal that's going to parallel that process where when you submitted a science-oriented paper from your model, you also submit to this journal, and you submit your code and your model and a description and documentation, and then their job is to vet it themselves, run the simulations, and test it out. And if your paper is accepted and they accept it, then they'll kind of go hand in hand out at the same time. But I think it's a good opportunity to have the normal peer review process, but then also have a peer review process on the validation aspect, and then get that all out there kind of together. So it might be something that MSM could start to be more involved with as he gets the journal off the ground.
Yeah, the other thing also I've always thought about is with the committee, at some point we're probably going to have a suggested guideline on like a standardized reporting method, basically. And then the other thing is, in addition to Peter Hunter's journal, there's the ASME VMV journal that just started. So that journal is specifically designed for accepting papers, describing verification, validation, and credibility assessment methodologies, which is directly tied with VMV 40 subcommittee that they have, which was credibility assessment of models for biomedical devices. So
I just want to follow up with Silvia's, comment, because we have recently done a study about introducing model sharing and reproducibility assessment to scholarly publishing, and that was actually the example study used in the poster on the credible practice, 10 simple rules. And I think that is doable, which, I think this might open other discussions too, but I think one thing we also need to resolve is to balance the burden and cost of these processes, with the necessity of doing those too.
Right. The peer review publication and Peter Hunter's efforts, they're all fantastic, but they address the outcome of a third-party evaluation, not necessarily expose the process as much, although even Peter Hunter's might get there a little bit. So it's important to know what actually didn't work, or what was the iteration, what was the learning if it actually occurred, as opposed to just, here is the best thing that we came up with after we did whatever we did, and we didn't keep track of, or we can't tell you what we actually ignored. So the negative result problem, right, so to speak, that's part of the learning that has to be documented in some way. I'm not discounting the value of the peer review or the run my code and all these Physiome journal things.
So, Raj, I think you make a very valid point, and I think part of the dissemination of your work should involve also various versions that you've arrived at, and also highlighting the errors and limitations of those different versions. And I think that would address some of those concerns. But one thing I will say is that even the most rigorously documented process, I can find things that are flawed with it. So I think we have to start somewhere and then build from there.
Right.
So.
Can I just follow up really quickly? That really relates to a lot of the discussions we're having at NIH when we're talking about open science and open data. And you learned about the SPARK program yesterday, and literally we were thinking about having legacy data and to have data that's documenting the negative results so that the community can learn for building devices, for collecting physiology, for building models. And so it's definitely an ongoing discussion and having all the results available.
I'm Ruger van Den from Stony Brook Applied Mathematics. Daniel Bluestein just reported the results took almost 1 million lines of C and MPI code and running on 10 to 100,000 cores. And I just noticed the first time I'm attending this workshop, people have a MATLAB and a Python codes. I don't know if they are pretty long, pretty firefight. So I have 3 proposals or 3 suggestions just following what Grace was saying. Number 1 is to have computer scientists to join the community for 2 purposes. The first one is to speed things up, calculation. You just mentioned that you cannot do a real-time calculation for trauma. If you put on a 1 million core machine, you could do a real-time. Of course, that's another undertaking. The second purpose is for the software engineering for validation and verification. So Grace just mentioned for community sharing of code. So it would be very nice to have this is my second suggestion, to have a community shared code. We don't have to everyone create a piece of module with a lot of overlap. So this is a second. Third one is the validation verification is indeed a multi-scale. You go all the way from science to the model and to the programming. Every level has to be verified and validated. So particularly for the programming part, you need to have computer scientists, even people like me in applied mathematics, to join the community. Yeah. So that's what I'm.
I'm curious. Are there computer scientists in the room? One. There were more. Yeah. I'm curious if people have. I'm at UVA, and we have a lot of discussions about how to integrate with computer science more. And there we have a little bit of a cultural issue with that, in the sense that, like for example, I've thought it would be great to have a PhD student in computer science work in the lab and do their PhD in our work. But the computer science department where we are wouldn't see that, kind of implementing algorithms in context of what we do as pure computer science. They really want hardcore new theory and algorithms that, and the kind of anything that feels like an applied, they don't want a PhD student. Even if that resulted in a Nature or Science paper, to them that's still not a PhD. So I'm curious if anybody else has had a similar kind of experience like that, and how they've addressed it.
Yeah, I've actually made the same observation in the past, where trying to get computer scientist students to work on applied engineering or medicine related work. It's like, no, they want to get into the core of computational method.
They're actually, the students are interested, but the faculty don't necessarily agree. Yeah.
So their schools actually discourage it because
Right,
their
yeah.
focus is something else.
Well,
So I can,
I've got a solution to that, but I don't know that you'll like it. So at National Labs, we work very integrated. We work with computer scientists, microbiologists, physicists, all in the same team. But the downside of that is that we don't have the graduate students, and they're not working towards their PhD unless they're working as an intern on the project. So even though we can get that very collaborative culture, it also drives the cost up. So I think there's a couple universities. I think UC Riverside is going towards this cluster model too, where you're teaming to do this interdisciplinary science. And we were having a discussion about this last night at dinner, that that hopefully you can still keep the university cost structure if you do that, and not have to go to professional programmers who are on staff, because then it makes that U01 project hard to be cost effective.
So I can mention that our team consists of, a major partner is the Pittsburgh Supercomputing Center. So we have a lot of software developers, computational specialists, and things like that who are intimately involved. So I did share your experience, Silvia, but I think there's a whole spectrum. So certainly a large percentage of people who are steeped in computer science, are more interested in sort of developing new methods and algorithms, et cetera. But I think there's an increasing amount that's bleeding into the transdisciplinary fields. So one of the challenges is finding those people. So I've seen many times where where teams have formed in the opposite manner, where you have some people who are maybe not modelers, and they say, okay, let's just throw a bunch of engineers or computer scientists at a problem, and that doesn't work out naturally, because the folks who want to do that, they themselves don't necessarily understand modeling, and then the computationalists aren't oriented that direction. So part of it is, I think, both, or I shouldn't say both, because I think there's multidisciplinary. You've got sociology, biology, computer science. You have people in each of those fields who are merging within this transdisciplinary field, which is emerging. And we have to understand that the whole field of multi-scale modeling, in many ways, is a field in itself. In other words, it's transdisciplinary, but you actually have, the field itself has to have a base understanding. People have to understand how to work with each other. There's a whole set of skills and abilities and orientation that are involved in doing these types of things. So I think the future is attracting more people to this area so that they can, submerge themselves in this broth of transdisciplinary approaches.
Sounds like it could work.
So following that, solution, and I'm sure that it's done in other labs, to embed graduate students from other departments, in our case, applied math, and we found advising students to that, where they actually see also the wet lab experiments, because this is how they will validate or test their simulations, and they can consult with the other graduate students who are involved in the other aspects of the simulation and the validation. So this is the true interaction that enables the real experimentation and validation of the model.
in terms of outreach, maybe this is a job for your new committee, a working group, should I say. We have, sorry, go ahead.
Also want to add to address your question. So we've run into situations where we had to develop new methods in the process of building a model. So especially when we're doing very large-scale models. So you essentially have, like if we build a U.S. model, that's 300 million plus agents. So by definition, you have to build, multi-threaded. So that changes the structure of the code. So those are computer scientist questions. So it was helpful to have that expertise on board where we could then just adapt because we were realizing, we were filling all the cores very quickly. Another example is visualization. So visualization is extremely important when communicating with decision makers. And we have to develop very hard-hitting, quick-hitting, but easy to understand visualizations that are interactive. And that whole process in and itself is also, you're developing new methods. So I agree with you. I think it's helpful, and this is a way you can iteratively really move forward all these different fields.
We have time for one more question.
No, just I think you've made the point that in some specific instances, there may be computer science going on, where it's helpful to have computer science students. But in general, the problem of evaluation is probably more a software engineering problem, and there may be less software engineering students than there are computer science students.
I just want to close up, like, how do investigators feel about this request for credibility plan? Do they feel burdened? Do they feel it's necessary? How, anxious they become? Or do they think the committee's activities are actually helpful or detrimental? Yeah.
At first it seemed very open-ended, which was, challenging given, the fast response time. But I think once there was some communication about what the attributes of the plan were, I think it was everybody came at it very different ways, but I think it prompted a lot of original thinking on our part in our group. So I think it was valuable. We'd love to see, supplements to budgets to hire people to do this.
I want to remind you that the cap was at $500K direct costs, specifically to allocate funds for the model credibility plan, outreach in the multi-scale modeling consortium, and activities.
On a more serious point, I could imagine a postdoc in F-32 computer scientist trying to write an application in this, and maybe not faring well in the review process because it's so interdisciplinary and it would seem kind of applied. It may fall between disciplines. So, anything to encourage people to follow career paths like that, very deep computer science skills applied to these large algorithms would be fantastic.
So I think it's part of the process already. So it's not necessarily added burden. I think the one thing to keep in mind is there are certain general principles, but then the model credibility steps and validation steps can be quite different for different models. So caution about the attempt to maybe have a one-size-fits-all, and that there's going to be certain things that you want to look for, but then there's going to be needed flexibility depending on the size of the model, depending on the purpose of the model, the intended audience, all those different things.
So on that note, that's actually a fantastic point. What we were hoping is that some of you would be actually willing to share your credibility plan and how it progresses over time, because it would serve as a fantastic example that we can share with the rest of the community to demonstrate to them that it's not a quick, you're expected to follow this one rigid path. It's a fairly flexible thing. It's not the destination, but rather the journey of demonstrating the credibility of your model.
I think one thing that would be helpful in the conversations, again, this is an outward-facing thing, as part of the dissemination working group, would be thinking about how to communicate. I mean, this is inward in the sense of we're helping each other define these things, but to me, one of the really utilities of it is to help communicate to the outside world what it means to be credible in doing modeling. So then we write, journal papers and grants, and it's not getting reviewers that just say, this is just modeling, is whatever. To have to think about how to really promote it to the outside and show that what we're doing is a real discipline, and it's got. So I think that's what we're talking about, but then thinking about what's the best way to communicate that to the outside world would be.
Yeah, right now one of the things that we're doing is actually holding things like webinars, where we do invite external people who are completely outside of the MSM community to actually show us what they've done, and we share their experience via like the WebEx and also recording of that WebEx. At least from the perspective of the committee, the intent is not just to focus on like this community, but rather to provide a methodology that's applicable to researchers across the board. But definitely the dissemination process can always improve.
Just a brief comment about third-party evaluation. So just for example, now a proposal that was written a couple of years ago, we actually took software company, Boston, to serve as a third-party evaluator, but recognizing evolving the modeling and also what we found out here through the consortium activities is that most likely third-party evaluation will be done better by reaching out to colleagues who do similar things and are well equipped in order to do this type of third-party evaluation. And we've already started such discussions among us. I think that this would be a kind of practical, more efficient approach.
With that, thank you very much, everybody. Sorry for being 5 minutes over time, Grace.
Thank you, Alam and Ahmed and Joy, for a great last session. And before we adjourn, I just want to remind you to look all around your seat. If there are any cups or anything that looks like food or beverages, please pick it up and bring it outside. We're not allowed to have it in here. We do want to come back here and visit again. I just want to take this final time to thank everyone for coming to this 10th anniversary, and I hope you enjoyed it. And I will be in touch with you to set the date for next year and get feedback from you for this meeting. As always, we want to continue to improve this meeting and make it useful for you. So thank you all again, and have a safe travel home.